All right, and as people come into the room, we will get going in just a couple minutes as everybody comes in. Great. Okay, so welcome everybody to another CCN Generative Adversarial Collaboration. My name is Megan Peters, and I am going to be the MC for today. And we'll start off with a description of what the GAC project is, just to get everybody on the same page. I am sorry if you've seen this before. If you've been to some of the other ones, this will seem oddly familiar, but here we go. So first we can start off by just telling everybody about the program committee for this year. So instead of this year doing a kind of traditional CCN, we decided to do something different. And this was put together by Nico Grigas-Corte and Gunnar Blom, who are the co-chairs of the program committee. And then myself and Ralph Hefner, Gemma Roig, Eric DeWitt, and Jennifer Lieberman, who is here with me today to make sure that everything goes well. So first, what is a GAC? What is a generative adversarial collaboration? The idea here is that science benefits from alternative ideas and theories. And often what's happening in science is that I have my theory and I go out and I try to test it and prove it. And you have your theory and you go out and try to test it and prove it. Then after we have both published our beautiful papers, then we end up coming together afterwards and arguing about who is right. And the goal of the GAC is to kind of flip this thing sideways. So basically to find folks who have alternative theories that make opposing predictions about a particular phenomenon and get them to actually collaborate to come up with theoretical or empirical approaches that might actually dissociate between these two theories. And so the goal of this kickoff workshop and the GAC project over the next year is to design theoretical or experimental approaches that will allow us to arbitrate the two or more theories that are being presented in the particular workshop. And of course, our goal is to advance science, but also because we think that this is a healthy and friendly way to actually go about making real progress in scientific inquiry. So there are also some benefits, not just for science, but for the CCN community as well, all of you guys out there in the audience. And so one of these benefits is that we have open public debate. This is a great learning opportunity for early career researchers. It really kind of puts everything on the table. You get to see people think and talk in real time and engage with each other in a way that's not just kind of unidirectional, like I'm just going to present my slides and never have a back and forth with anybody. We also have community engagement from all of you. So these workshops and the GAC project in general are meant to be really engaging and interactive. And so we really encourage you guys to make use of the Q&A box that's at the bottom of your screen or the chat. If you set your chat to all panelists and attendees in your little blue chat button, then you'll be able to engage with each other. And the chat also has this cool thread, or the Q&A box has this cool threading feature where if you submit a question, then you can actually reply to each other. And so hopefully we'll get some community engagement and good conversation going today. And not just today, but also throughout the GAC project. So we encourage you guys to stay informed and stay engaged through the publication of the position piece that will be coming out later, probably early next year. And then also those are, you have the opportunity to submit commentaries to those position pieces as well. And also because science should ask the important questions. So the GACs were all reviewed on open review. And so this is kind of a democratic platform to decide not only which questions are important, but which criticisms of those questions are also important. So this kind of crowdsourcing of science and scientific engagement is really going to move the field forward. And then finally, of course, this also goes with transparency. So not only do we have open review, we invite you all to these kickoff workshops, and then we have those position papers and also the commentary on those position papers to really highlight the community engagement and the transparency of kind of making progress on these critical questions. So this is our schedule overview. We've already gone through 3 of these and they have been fantastic. And we've got 3 more starting today. We've got generalization of information, integrative encoding, or category-based inference, and that is the one that you guys are in right now. And then we also have 2 tomorrow, dimensionality and flexibility of learning in biological recurrent neural networks, and grid codes for generalization and flexible decision making. But if you missed one of the first 3 and you really want to see it, we are in the process of cleaning up the recordings, removing pauses and bio breaks and so on. And those will go up onto our website in the near future. So watch out for a Twitter announcement from the CC Neuro or the CogComp Neuro account to know when those are ready. And those will be posted and linked on our GAC website. So most importantly today, we want you guys to have fun, and we want you guys to please participate, get involved, be engaged, use the tools in the chat and the Q&A, and raise your hand if you want to say something and follow up on a question that you ask, and we can hand you a microphone, or if you really want to at some point during the discussion, maybe even pull you on stage and get you involved. So hopefully we're going to have a lot of fun today, and with that, I will stop talking. And I will pass the baton to the actual folks you came here to see. So why don't we just take it away with our first category, which is Aurelio and Jesse. So thanks for being here, guys. All right, here we go.
Okay.
Thanks.
Hi. Okay. Hi, everyone. I'm Aurelio Cortese, and I'm part of this GAC on the generalization of information, integrative encoding, or category-based inference. So first of all, I would really like to extend a massive thanks to the entire CCN organizing committee for putting together this really new and I think exciting initiative and making this adversarial collaboration possible. I think all of us in this GAC, we're all very excited of today's workshop, but also I think of what lies ahead in terms of challenges, in terms of the discussions that I think this will lead to. And I think now is a good time to actually introduce the whole team. So we are 6 neuroscientists that are spread across 4 different countries around the world. Jessica Taylor from ATR in Japan, Dasha Zaitamova from University of Oregon in the US, Xiaoshuan Pan from the East China University of Science and Technology in China, Helen Barron from the University of Oxford in the UK, Masamichi Sakagami from Tamagawa University in Japan, and to close the loop, myself, also from ATR in Japan. We also have, in the end, quite different backgrounds. Several of us have experience in behavioral, human behavior neuroscience, but also neuroimaging. Again, some of us routinely do computational modeling, but we also have, for example, Pan and Masamichi that work with monkeys and electrophysiology, or Helen that works also with mice and optogenetics. So let me first kind of put out today's kind of overall workshop schedule. So we will have first kind of general introduction by myself and Jesse. And then after some questions, we will actually move to the core part of the workshop, which is what we call here the view, pitches, and the debate, where Helen, Dasha, and Pan and Masamichi will present their views and kind of really set the stage for an ongoing discussion. After that, we'll move to what we call the experimental proposal. So here, several of us will present ideas or new experiments that could actually resolve some of those controversies that we have come across and that we will try to highlight through this workshop. And then the discussion will start again based on those proposals, and hopefully we'll also continue towards a bit more of towards the end, a bit more conceptual part. I think really important point is that we would really like to hear from all of you, to hear your opinions, your comments, your questions. I think these are all very important for us. So please, we're really looking forward to hear from you. So today's goal, first of all, we'll define a bit of the working terminology. We'll try really to highlight what are the controversies in these ideas of integrated memories versus or category-based inference. Discuss different views and opinions. hopefully in a nice and polite way. Then also try to discuss experimental approaches that could resolve the controversies. And finally, perhaps try to find, are there additional open questions that we actually didn't think about that actually are important? And from there, go on for future steps. One thing that you will hear a lot in this workshop is really this word, generalization. And in its essence, it's really basically using previously learned evidence to predict relevant behaviors or outcomes in novel situations. Now, and I think this is really at the core and like an essential aspect of human and biological intelligence, this ability to sort of to adapt and to make optimal choices in situations that we either have never experienced before or that are at least partly novel. So if you really think like from the simplest possible example, for example, in the morning, and I have to choose, I want to choose whether I will wear a black or a gray t-shirt, to choices that are much more, that have many more ramifications, much more importance to life. For example, should I buy the red house or the blue house? So for example, I might decide to buy the red house because it reminds me of my grandparents' house. So my past experience, memories, and so on will come into play and help sort of make those choices when we are in kind of in a situation that we have not exactly experienced before. And so let's say, how is the brain actually doing this? So what are the possible strategies? And one is we can generalize our behaviors based on really on previous episodes or previous memories. So we can have, you know, we have memories of different episodes, and we have experienced different type of informations, and all of this might have bits of like there might be a bit overlapping, and this bits of overlapping information could form maybe actually a very simple rule for the brain to sort of pick connections and to link memories together into sort of more combined and more holistic memory that then can aid more efficient behavior in novel situations. But I think especially for us as humans, we really tend and we really like to think in terms of categories and concepts and abstractions, if you will. Now, again, to give you a simple example here, you have these random geometrical shapes with different colors. We can decide to sort of categorize them into based on the color or based on the color here, like light or dark colors, or we can do this based on their shapes. So also this kind of this idea of categories or concepts, also from this we can see that it can be sort of really deployed based on the demands, on behavioral demands. Now, these are kind of really general examples and so on. So when it comes to the lab, I think it's useful to also to give a kind of very simple example of a task that we could use in the lab. And also, how can we measure? What kind of behavioral measure can we use to say, okay, well, behavioral is generalized in this new situation? So we can look at both during learning, when we're learning new association, or when we're testing them later on in a really to test the generalization. We can use as measures of performance, we can use reaction times. We can use, for example, the % correct in choosing the generalized options, as we can call it. We can look at the sort of how one fares at the first time that a new trial comes up. We can even look at the degree of false memories formation. So you can think that you might have new information that you have never ever experienced before, but you might think that you have just because you have sort of overgeneralized. Perhaps you have linked it to something that you have experienced before. Now, if I give you a really simple sort of lab kind of type of experiment, you can see here we have like trying to associate images, like two groups of images. If you look here on the right, you have an apple, a carrot, a kiwi, and so on. And on the right side, we have scenery, a snowy scenery, a river, and so on. And so we might experience many of these pairs together. And if later on I'm going to show you, basically I'm going to ask you, like for the carrot, you might choose between those two options. You might choose the river because you've really experienced them in the past. So you might think, okay, well, I'll choose this one. On the other hand, for example, if I present you this spinach, And this spinach maybe you've never experienced with either of those two scenery, this field and this kind of cloudy landscape. So in that case, you most likely your brain will try to use previously experienced evidence to actually predict which one would be the best answer in this case. And if you think about it, there's actually one simple way to do this could be to link memories together. So you've experienced the spinach with the river in the past, you've experienced the carrot with the river, but you've also experienced the carrot with this field. So sort of the carrot kind of creates this link, and now you might be able to link those two separate memories, and then this might aid generalization. Sort of activating overlapping memories then can allow them to be integrated together and re-encoded in a new memory, essentially. But maybe some of you have realized that in this example, the fruits are always paired with the snowy scenery. And if you look at the river is always paired with vegetables. So you might think, okay, actually here you might actually use a different strategy, and you might extract sort of really use this more conceptual or categorical labels. And when you're facing this new trial with the spinach, you might think in the past, okay, well, this field was associated with a carrot is a vegetable, so this scenery is associated with vegetables. So in this case, spinach is a vegetable, so I should choose, let's say I should choose the field. So in this way, basically, we are trying to put items into logical categories, and then we can take information that is relevant for one category member. For example, we have here this field, and we can propagate it to other members. So we can take this field where it's a vegetable, the carrot, and then from there go to the spinach. So really, with this really simple kind of sort of lab thought experiment, we have the same behavior, but there are two potential underlying sort of strategies or mechanisms. So which one is more likely to be correct, or is there one that is more likely to happen given certain conditions, or are the neural circuitry even the same for the two sort of mechanisms? And if you look at the literature, the brain regions that have been mostly related to generalization span this sort of kind of circuitry that spans the sort of subregions of the prefrontal cortex, the hippocampus, and let's say the medial temporal lobes, but also the basal ganglia and midbrain. And so I think this is where sort of we can start to put a bit together, like a sort of where the controversy lies, and I think which Jesse will actually try to really walk you through and then try to find those points of controversies, because we can have individual or concerted contribution from different regions. We can have that either those two strategies might be deployed in parallel, or there might be some kind of arbitration that could depend whether this happens during learning or at the time of generalization. So when the behavioral demand is really kind of put on, or it might also differ based on whether we could use, let's say, really well-established kind of very well-established categories or this conceptual information, or something that is really newly formed. And with this, I think I can leave the stage to Jesse to continue with the slides.
I have it. There we go. But first of all, hang on. I just want to see if I can get rid of, they seem to have gone. There were some little weird things on the side of the screen. Okay.
And they seem to have disappeared now, so great.
Okay, then I will start. Hang on. Okay. So, although we often talk about generalization almost as if it's one thing, there's lots of different paradigms that we use in the lab and things that we do in real life. And it's definitely up for debate whether all of these really tap the same underlying neural processes, or even if there is one certain thing that underlies all of them that's in common, or whether we're just looking at completely different things. So I'm going to walk you through some of the different paradigms that we use. The first one that I'll talk about is called acquired equivalence. And this is where you might learn, so on the top left, you might learn that A leads to C and B also leads to C, and that A leads to D. And then you'll be tested to see if you can make a generalization of information to presume, therefore, that B also leads to D. another related way that we might look at this is associative inference. So this is where you might learn that A and B go together, B and C go together. And if you make a kind of inference here or generalize enough information, you might therefore conclude that A and C also belong together. Transitive inference is definitely another way that we generalize information, but actually there's quite a lot of different definitions of this, and I'm not going to discuss them all. I'll just talk about the one that we've got here. So in this particular type of transitive inference, you might learn that A goes to B, B goes to C, and therefore make the assumption that A therefore leads to C. And to give you just one little example of how we might test this in the lab, if you look at the picture beside associative inference, so you see this lady Jan in front of this house. Then later you see this man Dave in front of the same house. And if you can make an associative inference and generalize this information, then you might conclude that these guys both live in the same house, so they might be a couple. I'm having trouble going between slides. Okay. So I'm going to walk you through some of the data that's related to different paradigms. And I'm going to throw a lot of data at you, so I don't expect you to remember everything. So I first of all just want to kind of show you how these are structured so you'll know where to look. So on the top left, I'm going to show you the paradigm I'm talking about. And beside that, I'm going to highlight the parts of the brain that have been associated with this. Underneath this, I'll show you what kind of experimental subjects were used. So humans or monkeys or mice or whatever. And also on the left in the middle here, I'm going to show you about whether these results were examined or found during the learning phase or during the test phase. So keep an eye on these spots for the things that differ between the slides that I'm going to show you. So some really seminal work in this area was conducted by Shohami and Wagner in 2008. And they used the acquired equivalence paradigm in humans. They found that basically, so if you look at the graph on the bottom left, they're looking at percent correct. So for trained trials, this is like A to D, which they've literally seen. And for generalized trials, this is the ones that they haven't seen, but could make a generalization about. So for example, from B to D. And they looked at people who were good and not so good at generalization. But if you look at the y-axis here, you see that it starts at 50%, which is chance. And so all of these people were actually pretty good at generalization. Even the people who are classified as poor generalizers are still generalizing above chance. So she found that people were pretty good at generalizing. And then if you look at the response times for these generalized responses, particularly actually for the good generalizers, the response times were basically the same as the response times that they made to the trained associations. So what this means, or what they concluded that this means, is that making a generalization doesn't take any more time than just literally recalling something from memory. And so therefore, because this happened at the test, they thought, okay, they must have already made the association that's relevant between the B and the D before they came to the test. They must have made this during learning. And in line with these kind of findings, they also found that activity in the hippocampus during the learning phase, but not during the test phase, predicted generalization responses. And so they concluded that generalization arises from an integration of overlapping memories in the hippocampus during learning. So this is a cool finding, but Dasha from our group has tried really hard to actually replicate this. And she has, I think, 5 different, slightly different versions of this acquired equivalence paradigm with 190 participants in total. And basically she doesn't replicate the behavior at all. So all of the participants were pretty bad at generalizing in general. And they had a big response cost. So it took them a lot longer to make the generalized responses when they did make them than to make the trained responses. So, I mean, there's already some controversy just within the same paradigm here that whether or not the results are replicable. And there's no known results that we know of where they tried to replicate the neural imaging stuff with this kind of paradigm. The neural imaging stuff has been tried to be replicated using slightly different paradigms. So for example, using the associative inference paradigm. This is the one where they might find out that A belongs with B and B belongs with C, and then they have to make the generalization, therefore perhaps A also belongs with C. And Dasha's also done some work here, and she looked at the hippocampus during the learning phase, and she found that, so if you look at the graph on the bottom left, the dark bars are the situations where the participants would go on to make a correct inference, and the light bars are the ones where they'll go on to not make a correct inference or generalization. And what she found was that during the AB phase, when they first learned something, hippocampal activity would not predict whether or not they're going to make a correct inference. But then when they look at, when they start to learn about BC, which is when they, so this is when they can actually reactivate AB. So this is the time where they really could reintegrate these things and save them together in their memory. So the activity in the hippocampus at this time was predictive of whether or not people would make a generalization response. And this was during the first exposure to BC. But in later papers, she found that if you show them, AB together a lot and BC together a lot, that with repeated presentations of BC, the kind of activity that you saw in the hippocampus, if it decreased across presentations, then they were more likely to make a good generalization response. So here the x-axis actually shows decreases in neural activity. But on the other hand, on the second graph, which for some reason you can't see well, this shows ventral medial prefrontal cortex increases in activity. And they found that across presentations, this increased. And also across presentations, there was increased functional connectivity between these two regions. So they concluded that in the hippocampus, there's really rapid binding of the information, which with experience gets transferred to the ventral medial prefrontal cortex. And in another study, they also found that people with bilateral damage to the ventral medial prefrontal cortex are not able to make these kind of inferences. So it seems like the ventral medial prefrontal cortex is quite necessary. So hang on. So a different way that we can look at generalization is to look at transitive inference. So this is the one where, for example, you learn that A goes to B and that B goes to C. And the inference, if you make it, would be therefore that A goes to C. And Helen from our GAC has done some recent work with this kind of task. She did it with humans and with mice. And she found basically that during the test itself, first of all, hippocampal activity overall would be predictive of whether you would make a correct inference. And if you actually look at the neural patterns of activity, the activity in the hippocampus, the representations of the, basically in this terminology, the A and the B would become more similar. And also they would keep their temporal order. So the populations that did fire for A would fire before B. And they said that this was a prospective code. At the same time, during the test, they found the activity in the medial prefrontal cortex and the VTA would start to become more similar for representations of the X and the Z, which in this case is the B and the C. So therefore they concluded that the hippocampus is doing a prospective code, which is then translated to the medial prefrontal cortex to make the inferred outcome. And in the same experiment, they actually did optogenetic silencing of the hippocampus and found that this prevented inference. And in a completely different study using transitive inference in mice, it's also been found that if you lesion the medial prefrontal cortex, then they can't do this inference either. So the medial prefrontal cortex also seems to be quite necessary for this. So in this kind of task, obviously, you need to keep a few things in your long-term memory. You have to have learned that A leads to B and that B leads to C before that you can go on to make the inference. But in a different experiment conducted by people in a different lab, they took out this long-term memory demand and they basically showed everything you need for the inference on the screen. And when they did this, they found that the hippocampal activity and also actually the medial prefrontal activity was not related to whether they'd make an inference or not. And in this case, what they found was that rostrolateral prefrontal cortex was more indicative of whether somebody was going to successfully make an inference. So I've been talking about really kind of stimulus to stimulus associative tasks until now, but I'm sure you've realized probably because of the topic name, that also if we start to think about groups of stimuli, then we can also make generalizations. So whether or not something belongs to a given group, or whether because you know it belongs to a given group and you've learned something about another member of this group, you can also infer that information for this member of the group. And there's a few ways that we can look at categorization. So the simple way is to look at perceptual categorization. So on the top left, you can see something really simple like group 1 is lighter colored circles, whereas group 2 is darker colored circles. And if you show somebody some novel circles that weren't any of these exact colors, but very similar, you can probably predict for a lot of them which group they might belong in just based on their color. And Dash has done some work related to this, where she uses her robot chickens, as she calls them. And I think she'll talk about that a bit later. Another kind of category that we could form is what we call functional categories. So in this case, the different stimuli that belong within the same category don't actually have to be perceptually related at all. Instead, they might all predict the same outcome, or they might all require the same behavioral response to them. And in this task here, the functional category inference task, participants first learn to form some functional categories. Sorry. Once they've really learned and overlearned these functional categories, then they start to learn some novel stimuli that belong to each of the categories. After this, one of the members of each category is associated with either high or low reward. And the idea here is that if the participants have really learned these functional categories, they should be able to propagate this information without any experience to the other members of the same category and be able to predict reward for them based on this experience with just one member of the category. So I'll quickly tell you some more results related to these. First, the simple one, perceptual categorization. So in this kind of task, it seems that the ventral medial prefrontal cortex and the anterior hippocampus are important, and this is both during learning and at test. So in the first study that I'm looking at here, which was one of Dash's as well, she found that after people had learned these categories, these perceptual visual categories in this case, there was increased connectivity between these two parts of the brain. And furthermore, she actually found that these two parts of the brain seem to track category prototypes, both during the learning phase and at test. So when I say a prototype, I think this is where it becomes really related to generalization here, because a prototype is basically an averaged representation of all the stimuli within one group. So it's not even necessarily a stimulus you've seen, you've just extracted all the information from the different items and found the most average one or the most generalized one. And so in these studies, Dasha found that more generalized category information was represented in the ventral medial prefrontal cortex and in the anterior hippocampus. And this is from the learning stage onwards. To go back to the slightly more difficult functional category, and in this case, adding inference on top of it, this is work by Pan and Masamichi, and they found that, first of all, the dorsolateral prefrontal cortex seems to be very important for this. If you inhibit it with mucimol, then the monkeys, so this for time we have monkeys as experimental subjects, they can't do the reward inference at all. Or at least the first time that they get to make an inference, the first time that they actually need to do inference. After that, they can just do the response they did last time, which is memory. They also found neurons in this part of the brain that track category. So these are the S neurons, and they have very, very, very similar response to all of the members of the same functional category, to the A and the B and the C and all of the novel ones for it too. And there's different populations that represent different categories. And they also found neurons that represent the relationship between each category and reward. So there might be some neurons that represent the first category, but only when it's in a situation of high reward. And this really looks like these are doing category-based reward inference, particularly in these neurons. So based on these results, you could say that perhaps generalization is a pre-FC-based category inference that happens at the time of response. But of course, for this study, and also for the mice studies that I talked about earlier, I mean, these are very much region of interest based. You can't stick needles all over the monkey's brain. You have to choose in advance where you're going to stick them. And this is actually the case with some of the fMRI studies as well, I should say. So now that I've thrown all this data at you, we'll come back to where does the controversy lie? And of course, we talked about how there's potentially individual or considered contributions from the hippocampus and different regions of the prefrontal cortex. And it's very likely that different findings, even that I've shown you here, arise because people are really concentrating on different regions of interest and not really testing the whole brain fairly. And also it's very possible that even which parts of the brain we use depend on the paradigm. They're probably not tapping all exactly the same thing. Some of these results were found during learning, some were found during the time of the generalization response. But again, we're using different paradigms, and some of the studies only looked at one of these 2 time periods. How does this fare for new versus well-established associations or categories? Well, again, we've got quite different results, and one of the reasons for this could be because people or animals were trained to different amounts in different studies. So obviously for animal studies, you need to train them a lot more than for human studies. The human studies, they can come on and be over with an hour, the whole thing. But even within human studies, there's different levels of training. And then that means that the association or category that you're basing the inference on is far more new or far more well-established. And this could change everything up as well. So our group came up with some key questions, and I've tried to cover them by throwing all this data at you and telling you where the controversies lie. So the first one is generalization just a simple memory association process, or are higher order inference processes required? The second one is, do experimental tasks that are used to investigate generalization actually all tap the same underlying cognitive and neural processes? Or maybe even more importantly, I'm sure they all tap slightly different things, but do they all tap at least one thing in common, which could be, actually what we can define as generalization? Does generalization that's based on similarities in perception have similar or different underlying processes to generalization that's based on similarities in function? Is generalization based on well-established association in categories similar, or does it have the same underlying processes as generalization that's formed from completely new associations and categories? And in which cases does generalization depend more on neural activity that happened during the initial learning compared to at the time of the generalization response itself? So these are the key questions that we're planning to base this whole workshop around basically today. And I've finished the introduction now, so we'd like to open the floor and invite everyone to ask questions to the group. And yeah, thank you.
Great, thanks Aurelia and Jesse for a comprehensive introduction. So if you have questions, please go ahead and add them to the Q&A box and we can get started on the Q&A period. But before we do that, and while those questions start rolling in, I think it might be nice to have, if any of the other panelists have anything that they would like to add or say or present kind of, I know we'll have presentations of opinions and position pieces coming up, but if there's anything that we'd like to kind of highlight to get us going on that, now would be a good time for reactions from the panel. So what do you guys think? No reactions. Everything that Aurelia and Jesse said is just perfect. Okay. Well,
no, I thought that it was really nicely comprehensive, and it was. I just wanted to kind of tell the audience kind of the one part that they didn't mention, and was as we were discussing kind of all these questions, we found out that we actually do not have that opposing view, just because we are looking for, let's say integrative encoding at the time, evidence that people integrated encoding, which for a lot of my studies aiming to do. It doesn't mean that actually I don't think that plain associative memory explains a lot, and that we sometimes do this all on demand from very separate memories. So as we were going through and wanted to outline that controversy, we were kind of going closer and closer together, realizing we actually do not have that distinct opinions. So kind of Aurelia and Jesse had to rework the plan for the workshop a couple of times as we were kind of going through, realizing we actually can't have that argument debate, because it's not like any one of us has one position discounting the other.
But we can still design some experiments to tease apart what's going on. That's great. And hopefully later today, we'll have some interaction with the audience to actually get into that as well. So we have a couple questions here coming in, and hopefully we will have more as well. So please use the chat and the Q&A box if you'd like to ask a question. But we do have a first question from Anna Shapiro. And Anna is asking, are functional categories usually or always about reward outcome, or should we consider functional in a broader sense? So what we can do here is I can hand Anna a microphone. So Anna, you now have a microphone in your hand and you can follow up on this. So panelists and those who just gave a talk, what do you think about this question?
Pano Masamichi, would you like to answer this, or would you like me to answer it?
Okay, so for the second question, or maybe Pam will talk about this later. So because we found the categorical codes in the lateral prefrontal neuron, so they may be formed with a reward. So at least irrespective of reward, just by action or response. Yeah, this is my answer to the second question. So, and first question. Yeah, this is a very interesting question. So maybe we have to pursue looking for this kind of yeah, answer.
Great, thanks. Anna, did you want to follow up?
No, that's okay. It just seems that in general, these paradigms tend to use reward as the outcome, and the brain regions that are involved seem to be brain regions that might especially care about reward. So that makes me wonder whether it's actually, the fact that it's about reward or value might be really important, or whether there might be, like, I thought the way that Jesse set up the problem of having kind of superficially or perceptually different items that predict the same outcome, that could be more general. So it'd be interesting to think about whether there's something more general than reward there.
Great, thank you.
I'd like to add a comment.
Oh yeah, go for it.
I think it's a really good question. I think we would like to know as well. The studies that Pan and Masamichi did, and I have to admit that I've worked with them a lot, so I am biased for this. So the studies that they did always related it to reward, but in the learning phase, they didn't test the learning phase anyway, but the way I like to think of it in my head more is like, there are a lot of things, like I've talked to Dasha about this and she didn't, I think, agree entirely, but maybe she did. But so I like to think of something like fruit, for example. They don't look the same, a pineapple and an apple and a pear, but the way that you should respond to them is the same. So in a sense, functional categories in my mind don't always have to be related to reward, but we haven't tested other types of ones. And I think it's been pointed out that those ones do have more things perceptually in common than the ones which we use, which are way more arbitrary. So I think it's an interesting place to look at the overlap between these completely arbitrary ones and things like fruit or snacks, which don't look the same or even taste the same, but they have some kind of behavioral response in common or some kind of outcome in common.
Yeah, I think that that was actually where the disagreement was, that maybe we realized we call the same thing different names, and then we call different things the same name. And this was kind of where I felt like, other than the reward, which I assumed you kind of have to throw in if you are doing monkey research, that it may be just difficult not to. I felt it's more like a typical associative inference task, where it's really completely arbitrary association between A to B and B to C, and you are asked, are A and C related? So in some way, for me, that was kind of like, oh, the way I would think about it is more as an associative inference task than a categorization task. So that was some of the source of disagreement, but it was more like clearly coming from the different paradigms and different species where we started. So hopefully we'll manage to propose some research that would tease those apart. But thanks for the question, Anna.
Great. Okay. And we have one more question before we move on, it looks like, unless others roll in. So this is a question that came in earlier in the session, but I think is kind of related to what we've been talking about. This question is from Raju, and I hope I pronounced that correctly. And the question is Raju Bapi, and the question is, are memories stored independently of their behavioral consequences? So this is again kind of about functional consequences, but maybe there's a little nuance here to the question that has not been touched on yet. So Raju, I've handed you a microphone if you'd like to follow up on this question. And do any panelists want to address anything in here that hasn't already been said?
Yeah, thank you. No, I just had the same question that Anna put in a more direct way. Yeah, and that has been addressed. Or at least we hope by end of this session, we'll have some insight. Yeah, thank you.
Okay, fantastic. Okay, well, that's all our questions for now. But if you do have more, please, of course, go ahead and type them in the Q&A. So do we have any other commentary from the panelists before we move on to the next section? No? Okay. Well, why don't we then move on to our section 2, where we have some views and pitches and debate. So who is starting us off here? Is that Helen? Okay.
I think so, yeah.
Great. Okay, take it away.
Just share my screen. Let me know if you can't hear me.
Sounds good, looks good.
Great. And you can see the slides change.
Yep.
Yeah, good. Okay, so thank you, Jessie, for the intro. I'm just gonna move that. All right. So I'm gonna briefly try and talk a little bit about some of my own work, I guess, mostly, as a way to try and describe kind of where I sit in this sort of balance between thinking about integrative encoding versus category-based inference. And I'm going to be focusing on tasks that are probably somewhere in between what Jessie was describing as associative inference and transitive inference. So the tasks I'm going to be looking at are where A is paired with B, then B is paired with C, and then in the inference test, A, you're asking whether subjects can infer C from A. And I think in relation to the questions that were raised, in the task I'm going to show you, we're going to have a C that is both rewarding and another C that's neutral, so we can compare those two conditions with each other. So this style of task has been used by many different groups, both in humans and animals. And I think, yeah, there's a lot of lesion work and human imaging work suggesting that the hippocampus might play a key role. But there's also evidence, again, from lesions and from human imaging that medial prefrontal cortex, or OFC, is also playing an important role. And I should mention that this is at the test phase, so when people are actually asked to perform inference. And so I guess in this sort of brief talk, what I'm going to be asking is, okay, so what computation do we think is supporting inference at the time of test? And what can that tell us about whether people are performing category-based inference or whether they're using some kind of integrative code? And I think it's worth thinking about sort of the functional properties of these brain regions and what we know about them, because I think that can provide key kind of insight into what the code might be that supports inference. And in this talk, I'm mostly going to focus on the hippocampus because I think we understand that a little bit better. But I'll come back to the medial prefrontal cortex and OFC later on when I talk about future experiments. Okay, so the task I'm going to show you, as I already outlined, it's based on a sensory preconditioning task, and I've replaced A, B, and C with X, Y, and Z. So we've got auditory cues paired with visual cues, X with Y, then we've got visual cues paired with an outcome, Y with Z, and then in the inference test, we're presenting X and assessing whether the subjects can infer Z. And we've got two sets of cues, the orange set that includes a reward and a green set that includes a neutral outcome. And we conducted the study in both humans and in mice. And in humans, they're training in a virtual environment and the reward is monetary. Whereas in mice, they're getting a sucrose drop that's delivered to a dispenser, or a water drop if it's neutral. And yeah, I should say that in humans, it was a wood chip for the neutral outcome. Okay, so to start with, what we wanted to do is really try and identify whether the hippocampus is necessary for inference. So there have been lesion studies that have suggested that this is the case, but of course lesion studies don't have fine temporal precision. You have the lesion either before the animals start learning, which of course then muddles up whether or not any impairment is accounted to a deficit in learning or in actually being able to perform inference. Or there have been a couple of lesion studies where the lesion is done after learning, but again, it's not really clear whether the hippocampus is actually necessary at the time of inference or whether you've impaired some other mechanism that might support inference. So in our study, what we wanted to do is use an optogenetic method that has very high cellular precision and temporal precision. And so what we did was we injected an optogenetic virus, which is ArchT-GFP, into the dorsal CA1 region of the hippocampus. That's primary output region from the hippocampus. And we did this in CAMK2 Cree mice, which meant that the ArchT-GFP virus was only expressed in pyramidal cells in the dorsal CA1 area. So when cells are expressing ArchT, this means that when we shine light onto those cells, they will shut down or silence. So we implanted an optic fiber above that region of the brain, which we connect to a laser. And this then provides a really, gives us high precision over silencing those cells. So here you can just see the expression of the virus in that region of the brain that's shown in green because the virus has GFP included in it. And at the bottom here, you can see what happens when we turn the laser on. We get very dramatic silencing in the cells in that region of the brain. So using this approach, we decided to silence the dorsal CA1 just during the time when the tone is played in the inference test. So we're exposing the subjects to the auditory cue, which is X, and then we're going to assess whether they can infer the appropriate outcome Z, both when the laser is off and when the laser is on. And what we find is that if we look at the difference between our orange and our green cues, so our set 1 and set 2, we can assess whether there's a reward-seeking bias for the orange auditory cues, the ones that are indirectly paired with reward, relative to the green auditory cues, which are indirectly paired with a neutral outcome. So we compare the behavior on set 1 and set 2 trials, and what you can see is that when the laser is off, there is a significant bias for more reward seeking for the orange tones versus the green, but when the laser is on, that bias is no longer observed. And if you repeat the whole thing in a set of mice that undergo the same surgeries, but they have a GFP virus injected, so there's no ArchT now expressed in the cells, these mice don't show any impairment. So this suggests that the hippocampus is really critical for inference at the time of inferential choice. But of course, this kind of raises questions as to what the hippocampus is actually computing during inference. And I think it's important to think about what we know about the hippocampus more broadly. And I've sort of come up with 3 different key features of the hippocampus, which I think are relevant to thinking about how the hippocampus might support inference. So firstly, we know from many different rodent studies that there are place cells in the hippocampus. So these are cells that fire in a preferred spatial location of an environment. And so we can think of these as just representing space, but we might also think of these cells as kind of representing the different elements or the relationship between different elements or stimuli that they observe at a particular point in space. So we might think of these cells as, in a way, providing a conjunctive code. And then, of course, we also know from human lesion studies that the hippocampus is playing a critical role in long-term episodic memory. So this might inspire the idea that we have some kind of retrieval mechanism in the hippocampus at the time of inference. And the third feature of the hippocampus is that during periods of rest or sleep, we see evidence for replay. So during rest and sleep, the LFP in the hippocampus is characterized by shortwave ripples, which you can see here, these high frequency oscillations. And what people have found is that during shortwave ripples, you can see evidence for replay of a previous experience that a rodent has had. So if this rat is running along this linear track, the place cells will represent A, then B, then C, then D, then E. And so during real experience, you get this sequence of cells firing, but then when the animal is asleep, this same sequence might fire, but on a temporally compressed scale. And we can think of this as forward replay, but there's also evidence for reverse replay. So yeah, I think all these different features of the hippocampus are kind of important for thinking about how the hippocampus might support inference. And just to briefly recap on what the sort of different solutions might be to the hippocampus supporting inference, we can think of there either being an integrative code where you have kind of overlapping representations in the hippocampus, where A, B, and C might all be encoded by one population of cells, or AC might be encoded by a population of cells. And an alternative would be that you've got sort of separate representations of either A, B, and C, or the learned information, which would be A, B, and B, C. So at least in my mind, these are the sort of two ways that you might separate out the possible codes that the hippocampus uses to support inference. I'm just going to skip over these slides mostly. So yeah, we've either got this integrative encoding mechanism, and I think, yeah, I mean, it's plausible that you could have integrative encoding given the anatomy of the hippocampus. So people have proposed that the CA3 region of the hippocampus might provide an integrator, or you could imagine that activity during shortwave ripples might facilitate integrative encoding. And retrieval mechanisms, there are several different mechanisms that have been proposed. One of which would be that you kind of rely on the pattern completion mechanisms that are inherent to the hippocampus, and this might allow you to have recurrence within the hippocampus that would allow you to kind of pattern complete from A to C, even though the subjects never directly experience A and C together. But an alternative might be that you're just chaining together the learned information across different neural circuits. So you might have A reactivating a representation of B, and that then leading to a representation of C, potentially in another neural circuit. And this sort of chaining together of memory is what I have found evidence for in my own data. So I'll just give you a little bit more detail on that. So using the task that I already described to you, what we asked was what happens in the hippocampus when subjects are exposed to the auditory cues X on their own. Do we see any reinstatement of the associated Y cue or the Z cue in the hippocampus? And what we found was that both in humans and mice, we find evidence for reinstatement of the associated visual cue in the hippocampus in response to the auditory cue. So you can see these red and blue squares on the left. These show the sort of models that we had that we were testing against the data to look for reinstatement of the associated visual cues in response to the auditory cues, where we have high correlation in the activity patterns on the on diagonal versus the off diagonal when we're comparing the response to these auditory cues in the inference test versus activity patterns that we record in response to the visual cues. And we only observe this for correct trials, as you can see from the bar plots and the null distributions on the right, where you can see that there is this significant similarity between the auditory and visual cues for correct trials, but not for incorrect trials. So this suggests that the hippocampus is actually using learned associations to predict the short-term future, and we can think of this as a prospective code, where the subjects are able to kind of look ahead within their current spatial context using what they've learned. And what's more, this sort of prospective code seems to retain the learned temporal statistics of the task. So this is just an illustration, but in the paper we have quantified this, and we show that if we pull out cells that represent the different cues X, Y, and Z, we find that during the inference test, the cells representing X fire before the cells representing Y. So these plots on the right here show that when there is this increasing probability of the Y cells firing after a spike in X. So from this, we conclude that during inference, hippocampus seems to represent a prospective code that includes the learned temporal statistics of the task. So it's really relying on a veridical copy of memory in a way. And what we didn't find evidence for was representation of the inferred outcome in the hippocampus. And that's despite the fact that, at least in mice, we can see clear evidence that there are cells that are capable of representing both the reward and the neutral outcome, but they're not representing it when it's an inferred outcome. So which brain regions might represent the inferred outcome? Well, in humans, what we did was we sort of scanned through the brain, looking for where the inferred outcome might be represented in response to our auditory cues. And what we found was that the medial prefrontal cortex and the midbrain, which is a kind of putative dopaminergic region, are showing representation of the inferred outcome at the time of inferential choice. And I think, again, relating to Anna Shapiro's question, what we find is that these regions still represent that outcome if we restrict it to the neutral cues. So it's not simply a representation of reward, but it seems to be a representation of the inferred outcome regardless of the value of that outcome. So from this, it seems that inference is involving a retrieval mechanism that's really spanning across multiple brain areas, across neural circuits, if you like, across the brain. Okay, and then finally, I just want to touch on what the role of rest and sleep might be. So in our experiment, we wanted to ask whether activity during offline processing might actually help join the dots between different experiences. So what we're observing during inference is that we've got this chaining of X, Y, and Z, but is it possible that during periods of rest, there might be the possibility to form a shortcut between X and Z, the two stimuli that have never been experienced together? And so again, we identified cells that represent the different cues in our task. And then what we did was that we looked at what happens, or looked at the characterization between these cells during shortwave ripples, both early and late in our experiment. And what we found was that there is this increase in co-activation of cells representing X, Y, and Z together from the orange set. So this is the set that includes a reward, and that's not the case for the green set, the one that doesn't include the reward. And what's more, we actually see this increase in co-activation for X and Z cells independent of Y. So it seems like during shortwave ripples there is formation or increased representation of the inferred relationship, and that's specific to reward-related activity. We don't know yet what the functional significance of this is, but you might think of it as activity that might facilitate the formation of a cognitive map, so filling in the gaps, across information that you've actually learned. So just to conclude where I kind of position myself, I showed you that silencing the hippocampus impairs inference at the time of inferential choice. And given the physiological and functional properties of the hippocampus, we can hypothesize about potential mechanisms that might support inference. During inference, I showed that the hippocampus uses a retrieval mechanism to predict upcoming future experience, where we're seeing that X is predicting activity in Y, or cells that represent X are predicting activity in cells predicting representing Y. And this hippocampal perspective code is then translated into a representation of the inferred outcome in the medial prefrontal cortex and the midbrain. And finally, during shortwave ripples in the hippocampus, there's evidence for this increase in representation of the inferred relationship. So an increase in representation of two memories, or co-activation of two memories that have never been experienced together. And of course, this activity might play an important role in potentially building an integrated code elsewhere, or in training neural circuits beyond the hippocampus. Yeah, I can talk more about that later if people are interested. Yeah, that's all. Thank you.
Great, thanks, Helen. And it looks like next we will have Dasha, is that right? Great. So before we move on, I'll just say that we do see that there is one more question in the Q&A, and we will address it after the pitches are over, so stick around, don't go anywhere. So Dasha, go ahead.
Okay, can you see my slides? Okay, thanks.
Yes.
So when Jessie and Aurelio approached me about this generative adversarial collaboration, I couldn't say no, because I feel like this question characterizes essentially like 3/4 of research in my lab. And I view it pretty much the same as Helen, or verbalize it as a question between kind of, is this something that happens at encoding? Are we building this kind of complex knowledge structure as we go along, or is it something where we kind of chain associative memories on demand at retrieval? And I studied the question kind of in 3 different paradigms, in category learning, associative inference, and acquired equivalence. And across all of these paradigms, I kind of convinced myself that integrative encoding is definitely real. It happens sometimes, but it doesn't happen always. We sometimes do store separate memories of individual events, kind of the picture she showed from the Kumaran model, and just kind of compute the inference on demand at the time of retrieval. So I'll show you kind of the new questions that I am now asking. Is not really it's one or the other, but when is it one or the other? And are these 2 alternative routes to generalization, are they kind of supported by the same network of region, distinct regions? And what my current kind of idea is how this works, I'll illustrate on an example of my research in category learning, but it seems it may be working like this in other paradigms, like that associative inference that Helen was focusing on. So category learning is really convenient to answer this question, because this question whether it's retrieval based from separate memories versus is it some kind of integrative encoding mechanism map really well on 2 well-developed models of how do we represent concepts. So let's take a concept of a dog. So based on exemplar model, we represent the concept of a dog simply by the specific dogs that we've encountered in our life. And when we need to categorize a new animal, we kind of consult these specific representations. In contrast, the prototype model assumes that we somehow extract kind of the central tendency, the ideal or average dog that doesn't really exist, but is the perfect embodiment of dogginess. as Jessie was explaining in her intro. And then we kind of consult this integrated or generalized representation when we want to label new animals. And because these conceptual models have also well-developed mathematical computational versions, we can make kind of trial by trial quantitative predictions about what we should see in behavior and in the brain if people are using one or the other representation to generalize to new stimuli. So in a study that we conducted a couple of years ago, we knew that, despite kind of traditional multiple memory system view that assumed that categorization is hippocampus independent, we knew from other studies that hippocampus is involved. So to see kind of what representation people are using and what does the hippocampus do and what does it represent, we show our favorite robo chickens to participants and give them few examples of each category A and category B, and then scan them when they were categorizing new robo chickens. And then we were testing whether their behavioral and brain responses are more consistent with what the exemplar model would predict, what we should expect if they are consulting the specific examples of each category that we gave them, or if they instead extracted the generalized representation, the integrated representation of each category, the prototype. And what we found in the brain and in behavior is that most of what we saw was consistent with the predictions of the prototype model. So about 3/4 of the participants were better fit by the prototype model. And we also found kind of several regions, including ventromedial prefrontal cortex and anterior hippocampus, that showed signal consistent with the prototype model predictions, and we didn't find anything above threshold for the exemplar model. So now, do we think that we proved the prototype model right and ruled the exemplar model? So no, despite kind of this encouragement that, integrated memory integration works in categorization as well, I feel that it's more complex, as usually is. So there is at least one prior study that looked at prototype and exemplar model in categorization, and contrary to our result, they found primarily exemplar correlates. And then in our behavioral study, we found out that we can actually kind of push participants to be towards more prototype strategy or exemplar strategy, depending on the specific task we give them. And even if the categories are exactly the same, the specific kind of instances that we use to give them an idea of the category, kind of the selected training examples, play a big role. So if they have, if they are coherent and similar to each other in certain way, then we see more prototype strategy. While if they are less coherent, we see that participants seems to be kind of encoding the separate representation of each exemplar and then using more of an exemplar strategy. And in our latest study, at least in one phase of the experiment, we found that we are able to see both prototype and exemplar correlates. The prototype correlates replicated in ventromedial prefrontal cortex and anterior hippocampus, but we are also, when we pushed participants behaviorally towards a little bit more exemplar strategies, we started seeing also exemplar correlates in lateral prefrontal, lateral parietal cortices. So these dissociations between kind of these prototype and exemplar regions also kind of nicely map into how I was thinking about specific memory versus generalization regions in cortex before. So for example, we know from other literature that there are cortical memory regions in lateral prefrontal, lateral parietal cortices whose job is essentially to keep similar things separate. So those are regions that represent kind of these veridic memories and preventing interference between similar experiences. But we also have cortical memory regions where whose job is to kind of take similar things and kind of lump them together into a summary representation, such as schemas or semantic memory, and those are in ventromedial prefrontal cortex. And then I didn't talk about it, but also middle temporal gyres, where we also see prototype correlates. So it's pretty exciting that we are seeing kind of these two map relatively nicely to the prototypes and exemplars in categorization. So to summarize my view, I believe that we definitely have both of these routes to generalization. They both happen at some times, or maybe they happen both in parallel. They seem to be supported by distinct regions, but rather than true division of labor, like these are, you know, the generalization region we use for generalization and the specificity regions or exemplar regions we use for, let's say, episodic memory. I think that both types of representation actually can contribute to generalization judgment. So we do use kind of retrieval-based strategy from separate memories in addition to integrated memories. And then I didn't have time to talk much about hippocampus, but you heard it earlier. So one possibility is that hippocampus represent events at multiple levels of specificity. It can be along different subfields, or it can be along the long axis of the hippocampus, anterior posterior in humans. And both specific representation in the hippocampus and generalized representation in the hippocampus can both also inform generalization. So that's the story is complicated view.
Great, thank you. Okay, so our next speaker is it Masamichi, is that right? Or is it Pan?
Pan.
Pan,
Yeah.
Okay, great, yeah, that's right.
Can you see me?
Yep, looks great.
Okay. It's good?
Yep, looks good. Here we go.
Okay. Yeah, okay, I will start. Thank you very much for the organizer. So yeah, for my talk, the title is category based.
Sorry, can I interrupt for 1 second and just ask you to hold your microphone a little bit closer to your mouth? That way we'll hear you better.
Now?
Yeah, that's better. Thank you. Sorry.
Sorry. So
Great.
category-based reward inference, maybe in the prefrontal cortex. So maybe we know the prefrontal cortex maybe include information or category, maybe such as cat and dog category. So our question is so whether and how the prefrontal cortex can use category information for generalization. We call the category-based inference. So we design this task is for the monkey. So we design let the monkey to learn this kind of two associated stimulus associated sequence. For example, A1 is associated B1 and C1. E2 is associated B2 and C2. The monkey learned those association in this we call the double cycle task. For example, in this trial, A1 is presented first. Then after a delay, B1 and B2 are presented. Monkey has to choose B1, then C1 to get a reward. Through this double cycle task, the monkey learned the BCA sequence we called. After that, the monkey also learned the BCA sequence where B1 and B2 are presented as the first queue, and the CBA sequence where C1 and C2 are presented as first queue. So with the training of this kind of association, we expected the PFC neuron could categorize all those associated stimulus A1, B1, C1 into A1 group. So when monkey learned C1 is paired associated is paired a reward, and we expect PFC neuron could predict A1 is also associated with reward. This is we call the category-based reward inference. Based on this idea, we designed this kind of task. So at the beginning, the monkey is learned C1 is associated with large reward. C2 is associated with a smaller reward in the reward instruction trial. After reward instruction trial, we call the double saccade. Double saccade trial is followed. In the double saccade trial, a collect saccade to the C1. At the end of this trial, we will get a large reward. So we will ask monkey whether can predict the reward based on the A1 stimuli. In another block, the stimulus reward contingency is reversed. So we should ask the monkey whether can predict the reward based on this A2 stimulus. So we recorded the neural activity from prefrontal cortex when monkeys perform this task. So we examined the neural activity just in the first Q period and found 3 types of neuron in the prefrontal cortex. We call the stimulus neuron, S neuron, stimulus reward neuron, SR neuron, reward neuron, we call R neuron. So actually, we found the S neuron represent the category information of a group of associated stimulus, regardless of their reward value. For example, this is a stimulus S neuron. So all of the trial were classified into 4 conditions. For example, this is the A1 stimulus is A1. A1 stimulus with a large reward, A2 stimulus with a large reward, A1 stimulus with a smaller reward, A2 stimulus with a smaller reward. You can see this neuron, this S neuron show higher activity to A2, B2, C2 than A1, B1, C1. And this neuron also shows similar activity to A1, A2, B2, C2. So this neuron may be called category information of A2, B2, C2. This figure just shows the population activity of those S neurons. The S neuron maybe can discriminate the preferred group from the non-preferred group, but they cannot discriminate each item within each group. So we call the S neuron maybe include the category information, not the individual stimuli. SR neuron may be called the reward information specific to a category of stimulus. For example, this neuron, this SR neuron show reward module activity to A2, B2, C2. No reward module activity to A1, B1, C1. So we call, this neuron seems to code the reward information for a group of A2, B2, C2 stimuli. This shows the population activity. So we call it in the preferred reward condition, SR neuron maybe can discriminate the preferred group from non-preferred group, but not individual. But in the non-preferred reward condition, they cannot discriminate the preferred group from non-preferred group. So in the R neuron may include reward information regardless of visual property. For example, this R neuron showed very similar activity to activity pattern to all of these 6 stimuli. From the population level, R neuron can discriminate the preferred 2 reward condition from the first trial after the reward instruction in 1 block. It means that C1 is rewarded, indicator A1 is also rewarded. the property of SR and R neuron in the PFC maybe suggests that the PFC use category-based inference to predict the reward. So one prediction from this model is that when N1, the new stimulus, one new stimulus is learned to associate with B1, and then the new stimulus N1 is added to this A1 group. Once C1 is paired associated reward, we expect prefrontal cortex neuron maybe can predict N1 is also associated with the reward. To test this prediction, we trained the monkey to learn this kind of new stimulus association. During this learning period, we use a symmetrical reward schedule. So after learning, there's no any asymmetrical reward information associated with new stimuli at this point. We let monkey learn a group of stimuli associated with B1, another group of stimuli associated with B2. Now, in the double circuit trial, newly learned stimuli are presented as the first cue. So we ask the monkey whether it can predict reward based on this new stimuli. Actually, we found that so S neuron can encode the category information for new and older stimulus. SR neuron may code the reward information for category for old and new stimulus. R neuron or SR neuron predict the reward value of new stimuli before their direct experience with a different amount of reward. So here shows the population activity of R neuron in the lateral prefrontal cortex. So PFC R neuron can discriminate the two reward conditions both from the first trial, both for older stimuli and the new stimuli. Especially in this trial, in this first trial, new stimulus have not been associated with reward asymmetrical reward directly, but PFC neuron still can infer the reward. But I just show one activity in the stratum, but we found that the stratum activity cannot predict the reward based on this new stimulus. So maybe stratum cannot use the category information to predict the reward. So we also found the muscle injection into the lateral prefrontal cortex impair the performance in the first trial of new stimulus. In this cell line session, the collecting rate in large reward trial is significantly higher than the collecting rate in the smaller reward trial. But in the muscle session, the two collecting rates are similar. So those data just show in this trial, in the first trial, we called some in the repeated trial, but the collecting rate in the large reward trial still significantly higher in the smaller reward trial for both the cell line condition accession and the motion concession. So based on those data, our conclusion is that PFC neuron may represent category information and the category level reward information to infer reward. We call the category-based reward information inference. Okay, thank you very much.
Great, thank you. Okay, so now we have an opportunity to have a conversation, and we have a few questions that have come in the meantime. So the first question that we have here, well, first, actually, maybe a good opportunity to have the panelists respond to the positions, anything that you want to argue against or follow up on. So yeah, opportunity to respond here, and then we'll go to the Q&A.
So, I mean, from my little perspective, the fact that there is that response on the very first trial anywhere in the brain, really, that already differentiates the two conditions for the new stimuli that they haven't experienced reward on. I know that you are scanning at retrieval, but that also could be used as an argument that this must have been already computed. It's already there at that first trial, so it must have been already computed. Therefore, to me, that sounds almost like a integration evidence. I think, is this the same thing, your paradigm, as what kind of was called in Pavlov conditioning? It's sensory preconditioning. So you learned that association between stimuli, then you learned that one stimulus is paired with reward. And then like I feel like it has been essentially used since Pavlov's dog, although, you know, not at this large scale of so many new stimuli, but yeah, but we are talking about, you know, having different names for the same things. I'm wondering if it's the same thing as sensory preconditioning, I guess. And then my other question is, do you see it as evidence that it, you know, that information must have been integrated and encoding because it's there, the first retrieval trial already?
Yeah, actually, sorry, can you hear? Oh,
Yes, we can hear you.
oh, okay. Yes, I think, yeah, according to my current the task paradigm, yeah, it's very similar as the preconditioning task. Yes, I agree. But the point is, so precondition maybe is just kind of the behavior paradigm. We don't know the animal or human, so the neural mechanism, how to perform this kind of precondition. Maybe just use our first categorize, then make a prediction. Maybe just kind of as previous suggested, so just kind of integrated association. So this may be just from precondition theorem. We cannot de-associate it. So this is category based or kind of association based. But I want to say, why I want to say this categories maybe mainly from some neuron representation in the prefrontal cortex. We found many category, pure category information just through this kind of association. So if we just this kind of integrated coding, so we don't need this kind of category representation. This may be my idea. Another we found so the SR type neuron. It's very interesting to combine this kind of information with the category information. So this maybe it's if just for example, if actually I didn't show some kind of results because so they just give me 8 minutes for presentation. Actually, we found some kind of if we found some specific stimulus reward association in the stratum without any category. So I think this may be different area, maybe have different representation with different mechanism to solve the same task, I think. And of course, maybe I should ask Masamichi may have some kind of comments for.
Oh, yeah. So I agree with you. But so for the sensory preconditioning, we don't need to use the category to perform it. But in a behavioral result with the neural result, maybe prefrontal neurons use the category to infer the outcome, particularly in with the new stimulus. So interesting thing is, so we looked that kind of activity in the stratum. So stratum neurons could predict the outcome. So when we used all the stimuli. So, but we couldn't find any evidence for the categorization in the striatum. So maybe although we didn't find the direct evidence, but from those results, we guess. So at least for the prefrontal neuron to predict the reward, maybe they use the category, we think. So we are now trying to show the direct evidence. So in the new experiments.
Can I say something? I think it's a really interesting idea that, and I think one of the people who commented on our reviews said on our proposal, they said, can we use integrative encoding to make categories? And I think that this is something we should consider. So we start to accumulate related memories until some level where they might become a category. But at that point, we really are talking about the same thing. But at that point, also, how do we determine, which stimuli we accumulate together and which ones we reject? Because otherwise we'll end up associating everything in the whole wide world with each other. And so we need some kind of rules that are going on. And I think that when we talk about category and then other people might be talking about inference, I think we're just trying to really emphasize that there's some kind of structural thing happening here where you're not just associating everything that you've seen together in any situation. And I think this could be a really interesting point, because I think that integrated encoding could be a way that categories are formed, but there needs to be some kind of rules behind it. Yeah, that's my comment.
Maybe I can ask Pan, like, how would you define an integrated code and a category, and do you think of those as the same thing?
Yeah, this is a difficult point. We discussed a lot. So actually, as Massimo suggested, we need further evidence to say this is a category, how to form the category. Actually, with just the current data, I cannot deny. So maybe such kind of some kind of integrated coding information maybe from the hippocampus, then transfer to the prefrontal cortex to form this kind of category. So for example, form this kind of information. So this point we should this is as just suggested. So we in the monkey study we cannot record the many areas together. Actually, in our task previous, we just recorded the prefrontal cortex and the stratum. We clearly show prefrontal cortex can use a category, show some category representation, but a stratum not. But I don't know whether hippocampus represents this category information or not. Or one possibility, so maybe hippocampus also can represent this category. Another maybe just make such kind of integrated association. Then this association project to the prefrontal cortex to form category like, type of like, but I don't know. So this maybe Jesse's idea. Maybe they want to check the whole brain, whether we can find more difference between the prefrontal cortex stratum, prefrontal cortex, hippocampus. So from this sense, yeah, but just the current data, I don't know good idea to answer to clearly separate this is category based, this is integrated integrated information coding. But the point is, so if we say this is integrated coding, we don't need the category representation. I only say this way.
So I think that I kind of see it the other way, right? Like I'm kind of on with Helen that the dissociation is this encoding mechanism where you form some kind of a representation that is linking these things already, versus is this some kind of retrieval based mechanism where you just code, store the separated memories and then kind of query them and chain them until you get from A to C or D. And from my perspective, kind of what you call as category-based inference is actually falling more into the encoding mechanism, like you already made some kind of link, and this is already a coherent thing. It's already a thing. It's not like just collection of individual association. It's somehow a thing because you have the representation, kind of the joint representation of all of them. There is that one neuron in the prefrontal cortex that just says this is category 1 and this is category 2, rather than kind of the separate code. So that's kind of where I see that, where this, right, like whatever you pin against each other, I see as the same thing, and the dissociation somewhere else. Oh, I mean, that's my view. But maybe the integrative encoding, you pin it against it because you felt like that's a loaded term which assumes certain mechanism and certain brain regions. So that may be another reason why, right? As in, you think that it's a hippocampal base versus prefrontal base. So maybe that's kind of why we see the dissociation in a different place.
Yes, I agree with this point. So in some sense, in our monkey study, we well train the monkey, and so in some sense, monkey already form the category. We don't care monkey how to form the category. We just monkey, we train the monkey, maybe monkey can form the category. Then maybe we want to ask them how to use this category information. So this is the point. So yes, I think mathematics maybe now he tried to answer. So how during the learning period, maybe how the neuron responds. So for this sense, yes, maybe I don't know. Maybe one guess is, for example, this kind of integrated coding for the mechanism. Maybe it's helpful to learn to form the category itself. Maybe from this sense, the category inference and the integrated coding maybe have different process in different stage. Maybe have different meaning for to use. So in some sense, we cannot compare them together.
So may I? So yeah, I'm very interested in how to form the category, particularly in the prefrontal neurons. So I love the hypothesis in a hippocampus. So category is formed. Then output is sent to the prefrontal cortex. Then prefrontal cortex uses the category to infer the outcome like that. But one thing we have to consider, so in the lateral prefrontal cortex, this area is located just before the motor process. So maybe prefrontal cortex receive the feedback information from motor related areas. So maybe, category, particularly in the category in the lateral prefrontal cortex, can be formed through the feedback of the motor output. So we know the one interesting series of experiments done by the Jun Tanji and Hoshi. So they used the simple task. So using a monkey. So they asked the monkey to use sometimes right hand, sometimes left hand to touch left panel or right panel. So then they looked activity pattern throughout the frontal cortex. So prefrontal, premotor, the primary motor area. So of course, primary motor areas neuron code the position and which hand monkey uses. But in the prefrontal cortex, the information is very abstract. So irrespective of the using right hand or left hand, if monkey touches the left panel, so neuron fiber like that. So maybe from the lateral prefrontal to premotor to the motor. So that some abstraction, maybe abstraction process exists from the primary premotor to the lateral prefrontal. This can be a hint, I think, to consider how to be formed, the category is formed in the lateral prefrontal cortex. So in that sense, I don't know whether if lateral prefrontal and hippocampus both can contribute to forming a category. I don't know whether they use the similar ways or not.
Great, thanks. This is a fun discussion. So before we take a quick break here, why don't we do just a couple questions from the audience? So we had some questions come in over the past session. The first question here is actually we had two questions from Ian Ballard. And Ian is asking, what about the role of attention in learning? Can they all be implicit, or do they require directed attention? And then we also had a question from Ian about the overall similarity of the stimuli shift and balance between integrative category encoding in the hippocampus and exemplar or memory-based codes. And so what I'm going to do here is I'm going to hand Ian a microphone. And I know that Dasha already addressed one of these questions, but maybe we can do these together and see if, Ian, you have any follow-up points that you'd like to make.
Yeah, I guess based on the discussion, I'm more interested in my second question now, the overall similarity of the stimulus. Because the way I kind of think about it is like potentially the PFC and hippocampus are two separate routes to category formation, like analogously to the ventral and dorsal visual stream. And where they might differ is like how biased they are to integrate across experiences versus distinguish similar experiences. And the hippocampus may prefer to distinguish similar experiences unless you hammer it with things that are just so similar, it's hopeless to try and remember differences between them explicitly, like robot chickens, in which case it extracts category structure, whereas the PFC, potentially because of its connection to motor output, may be biased to link across differences.
Yeah, I don't know if anyone else wants to answer this, but I was just going to say I would agree with that. And I think also if we look at like, you know, what the cells are representing in those two brain regions or what the structure of those two brain regions is, in medial prefrontal cortex, there's so much evidence, or in lateral PFC, I guess, as well, there's lots of evidence for sort of multiplexing and cells that are responding to multiple different stimuli, which would speak to an integrative code or a category code. Yeah, whereas in the hippocampus, there's like strong evidence for pattern separation, and yeah, that obviously would support a sort of separation of information. So yeah, I would agree with your kind of summary of how those two brain regions might differ in terms of how they're contributing.
Any other follow-ups?
I think though, doesn't Dasha's data, yeah, maybe Dasha, you should say it, talks about where it is within the hippocampus and stuff like that as well, right?
So yeah, I think that at least within the categories, that makes a lot of sense to me, this proposal, because it seems really at least we are able to push people towards more exemplary versus more prototype representation. And it's just difficult to even form the prototype. My only, just kind of thinking about kind of my prior life in associative inference world, I kind of feel that maybe it's not perfectly, does not perfectly explain everything, because the hippocampus was actually, I mean, I found the hippocampus to be integrated in that associative inference paradigm. The kind of the same that Helen is using, kind of A relates to B, B relates to C, and now let's see if A relates to C. And those are totally arbitrary associations. So it's, again, there is definitely plenty of retrieval-based strategies in that task, but there are also hints that maybe sometimes this is possible. I think maybe the question, it can be combined with the last Nastanich's point. I don't actually claim that this is computed in the hippocampus. I am perfectly happy to be convinced. I have this, so my only tool is MRI, so I don't have the single cell kind of timing resolution. I am perfectly happy to be convinced that kind of it's an interaction, let's say, with ventromedial prefrontal cortex that allows the similar things to be kind of linked together, and then the hippocampus kind of accepting the, you know, integrated memory. So I'm not kind of claiming the direction, and that could maybe be an answer to your question, where maybe the hippocampus always try to separate, but in some cases it can kind of more easily work with other brain regions to actually end up integrating or not integrating. And the other thing that Jesse was reacting to, it is also like we probably don't have a single engram for an experience. So right now it looks, at least in my hands, that maybe we are making multiple copies of every experience, and we have those separated representation, and we also have the integrated representation, and they are all in the, like they are in the hippocampus, and potentially they are there in the same time. I don't know whether it's like either or thing, or whether they can happen in parallel. But you know, it can work along subfields, and it seems to be working along the long axis of the hippocampus. And we know that when you represent space, right, there are these very detailed representations in the posterior hippocampus and very kind of coarse representations in the anterior hippocampus. There is a gradient with respect to kind of place cells, the receptive field of the place cells. So maybe hippocampus kind of does both, just to different degrees. But again, I'm perfectly happy to be convinced that it's the prefrontal cortex that helps the hippocampus to form the integrated representation at the end.
Great, thank you. And thanks, Ian, for the great question. So we have a couple more that have come in, and then we'll take a short break here. So the next most upvoted question actually is a related question, it looks like. This question is from Achiel Fenneman. And Achiel is asking, would the frequency of observations matter for when the prototype versus exemplar models may be more applicable, i.e. being a lower number of highly distinct stimuli versus a large number of rather similar stimuli? And unfortunately, Achiel, I cannot hand you a microphone. I'm going to have to yank you up on stage, actually, because it says you're using an older version of Zoom. So hold on for 2 seconds while I kick you out and bring you back in. Because the technology does it this way. There you go. So if you'd like to follow up on this question, we can do that.
Yes, this somewhat relates to some work I've been working on in the last few months, which is based on value-based decision making. So it's not entirely related, but then it does share some overlap with this prototype versus exemplar model stuff. And what I'm wondering is, I can imagine that if you have, for example, a number of very, very distinct stimuli, things that don't really look alike, but I also haven't seen it that much, that maybe makes it a lot easier for the hippocampus to draw out single memories. And then if I see maybe some, so for example, like the robot chickens, maybe I see a lot of these, that's somewhat related to what Ian said. Maybe if I see a lot of these very similar models, and over time I sort of like extract gradual co-occurrences and sort of use that to create a more well, statistical model in that sense. So I wonder if that's something that, I mean, this is just a hunch of me this time. Is it something any of you can weigh in on? Is this somewhat reasonable?
Sorry, I realized I talk a lot, but I've done a lot of research on this specifically. So yes, I think that that's a really good hunch. That's what we kind of noticed when we are looking at all the old papers, that behavioral papers that claimed, oh, prototype model is the best. They used kind of few highly distinct stimuli. And then the papers that show no, prototype model is the better, use larger number, kind of more coherent stimuli. So what we did was kind of use the robot chickens kind of across the board. The actual categories were the same, but we varied kind of the training exemplars to be either, you know, more distinct or more typical, and varied their number. Surprisingly to us, the number of training exemplars didn't matter as much. It mattered a little bit, but it didn't matter that much. The coherence mattered a lot. So when they were kind of near the prototypes, and the stimuli that I was showing, for example, there is actually no, you can't do unsupervised clustering and get the answer. All the training stimuli are still differing by, let's say, 4 features from each other. So which way kind of you slice the space, you have to learn via the task. There is no kind of natural, you know, more coherence between certain stimuli, but at least they are organized. Once you learn it, they are organized around the prototypes, and you can extract the prototype. Once we go to like 3 features away from prototype, then people move towards more exemplar strategy. But they tend to have slightly better recognition memory for the individual old exemplars. I mean, overall, because it's like a conjunction of 8 features, they have really horrible recognition memory in general. But when we see a little bit more exemplar strategy for categorization, we tend to see also a little bit better recognition memory when we test also kind of this memory for specific exemplars. So your hunch is very, you know, supported by evidence. Just the frequency was much less important in our hands than kind of how coherent the exemplars were. But overall, this seems to be the case. Did I answer your question?
Yes, I do have a follow-up question, but I don't want to like overly hog the time here.
I do think
Sure.
actually, unfortunately, sorry, unless it's very quick. If it's very quick, is that okay?
Very quick then.
Okay.
So I'm wondering, one final follow-up question: is there any sort of like over time as subjects need to task more? Would there be any evidence of subjects and switching from a prototype to an exemplar model?
You always switch from exemplar to prototype. So that's actually like Posner and Kiel who essentially established the prototype model. One of their seminal papers, 1970, Posner and Kiel, actually looked at kind of the strategy right after learning and then 1 week later. And the 1 week later, you see way better use of the prototype strategy. A little bit like Helen was showing about the sleep-related kind of seem to be happening, sleep-related integration. So behavioral that seem to be working.
So once you have a prototype, so once you have a prototype, then basically there's almost like no way to deconstruct it. So once you have this kind of prototype representation, then you have to stick with it, even if it becomes sort of, I don't know, behaviorally irrelevant, or even rather than irrelevant, even kind of counterproductive, let's say, to your behavior, you would still keep this prototype representation.
Yeah, you only increase you in general have a lot of false alarms to prototypes even immediately after learning, but it only gets kind of worse this time, or better if generalization is what you want. Yeah.
Great. Thank you. Well, I'm actually going to stop the Q&A here because we've got some other ground to cover, but we want to take a very quick bio break. So we will be back in 3 minutes, please, 3 minutes only. And then we'll be back to talk about experimental proposals. Okay. So I'll just put up a brief break slide here and we'll see you back here in 3 minutes. Thanks. Great. Okay.
Can you see my screen?
Yes, we can. And do we want to wait for anybody else or shall we get started? I think that was 3 minutes. I want to make sure we get through the experimental proposals. So why don't we go ahead and get going if you're ready?
Okay.
Okay, here we go.
So, so kind of to reconcile some of the discrepancies that we've seen, I would like to propose a couple of experiments. I'll try to be really quick with them. So my neuroimaging proposal is actually addressing almost all of the key questions that Aurelia and Jesse outlined in the beginning, especially the one whether generalization based on similarities, perceptual similarities, is the same mechanism as the one that is based on function. And just to remind you, kind of going from which model is right, I switched my question to when do we use each strategy, and because I found out that behavioral and brain mechanisms can vary across different category structures. So we can push people towards kind of more the integrative strategies, prototypes, when we provide coherence in the experiences, and or exemplar strategies, kind of the separate memories when there is a less coherence, and these are supported by distinct regions. So what I really think we need to do in order to answer this question is not to run two kind of studies or two tasks in one lab, but actually 3 tasks in one lab. We need to run perceptual categorization task, which we know, biases people towards prototype strategy. Another version of kind of very similar perceptual categorization task, but one that we know biases people towards exemplar strategy, and then run kind of the functional categories task. And we need to run this obviously in the same species, so we can kind of all look at the same regions at the same time. I would of course run the fMRI in humans. And what my hypothesis is that the distinction is not really between perceptual versus functional categories, but instead between kind of the perceptual categories that are based on summary representation, on that integrated prototype, versus those that are based on separate memories. So this is kind of how I imagine this particular experiment would come out. The answer would be yes, but it depends on what kind of perceptual categorization task you have in mind. I want to also propose a second experiment, because I feel like before we even go into and shell out the money for neuroimaging, we actually need to shell out money for a really long and complicated behavioral experiment to even set up the ground. So the second experiment I would like to propose is really addressing mainly the 3 questions, but focuses on the first 2 the most. Is this all just associative memory, right? I'm convinced that just the plain retrieval-based mechanisms from simple separate memories explains a lot. How much? And then the second question, do previous experimental tasks actually measure even the same thing? And to answer these questions, I think we need to run a large-scale individual difference study that would include kind of all the popular generalization tasks and also kind of simple associative memory tasks to so essentially it would allow us, are all of these tasks that we've used so far actually measuring the same thing? We've always cite each other and assume we are measuring the same thing, but we don't really know. And things that were thought to be completely, maybe even hippocampally dependent, like categorization, and definitely something completely different than, episodic inference, maybe actually tapping into the very same memory integration mechanism. So we definitely need to kind of have a study that includes all of these generalization tasks to see, are they really related to each other? And again, because memory for specific associations explains a lot of variance in generalization, we also need to toss in plain associative memory experiments to find out, you know, is it all just associative memory? So essentially what the large scale individual difference study would allow us to do is see kind of what, if people are good at one, if there is really truly something that is, you know, generalization above and beyond associative memory, core generalization factor. These generalization factor tasks should correlate with each other above and beyond what kind of all memory tasks have in common. And so doing study like this would allow us to see, like, are the generalization tasks actually measuring the same things? If not, do we have like two types of generalization, or are some of these really more specificity tasks with who knows what edit? To what degree this is above and beyond plain associative memory. And finally, if we really do have this X factor, the generalization above and beyond, what are the best tasks to measure it? So that's my pitch for the experiments that I think we need to run.
Great, thanks. Any comments from the panelists? And also we could do some questions. If you have specific questions about the experiments, we'll save other general questions to the end.
Yeah, maybe I can just ask one question about the neuroimaging experiment, especially about the prediction, you know, for the difference, you know, where you put sort of the line for the difference in the perceptual and functional, and more in prototype versus sort of exemplar based on. So, but I'm wondering in a way, because humans use a lot of, you know, semantics, we use words and so on to create those categories. So even in a functional way, if you get this kind of functional categories, in a way you could have the same kind of prototype, I would say. And if I think of the example of the dog, I think that you presented before, you know, I think in your introductory slide, almost if I have to think of you know, of my prototype dog, I would almost think of like a specific dog, and even an image of a dog that I've seen somewhere, rather than a sort of like an, I don't know, smoothed or average version of multiple dogs. So along that line, not sure, but it could also be that you would have this line, rather have perceptual kind of exemplar on one side and more prototypes and functional on the other side. Could it be?
That's an interesting thought. Well, it's an empirical question, so it's another reason to run the study now, because there is more than one reasonable prediction. Yeah, I think that the word prototype is actually used in both of those senses. One is the average and one is kind of the perfect. I think we've had that conversation about the fruits and vegetables. So you kind of choose one specific stimuli that stands for the whole category. Yeah, so that is definitely something that I can't rule out.
Great. Any other comments or any specific questions about this pitch?
So maybe I have one question. So here, just use maybe related. So you separate maybe perception and the functional perception category and the function category may share some similar mechanism. So this whether it depend on some species. For example, for the human, maybe we can use some language, maybe to describe the function and the perception. Some, for example, dog, maybe you have some category, some language meaning. But for example, for the monkey or for the rats, so the perception maybe and the function maybe is very different. Maybe they have no this kind of meaningful language to describe the function and the perception. So they just based on the similarity or stimulus itself. So I don't know whether this kind of mechanism, whether it would depend on this self or not.
Mm-hmm. Yeah, and I think that actually was one other thought that I had towards earlier and then I forgot, and it relates to some other questions in the chat. But yeah, I think that because you're like in monkeys, the functional categories are so overlearned, kind of as far as I would say as a human memory researcher, that the mechanism may be very different from what I would find if I run this in humans and had, you know, a few trials, because probably this is not a single trial learning type of paradigm, but just having a few trials, they may, it may really not be the same. I may not find the same mechanisms as you are finding in those really overlearned, overtrained associative tasks, where they may be building actually something like, you know, truly functional category from, you know, this is good and this is bad, more juice, less juice. And maybe there is no other way to recreate it in the lab with humans than to actually offer, for example, money, or making, you know, like maybe abstract category label like A and B is just not going to be enough unless I would, you know, do this for weeks. Yeah, so that is a good point, that maybe some of the differences that we are seeing are simply because of the different species and kind of other tools that we have available as humans and that they don't have, and the paradigm that we have to run them differently and you have to overtrain them.
Great.
Thank you.
Well, I think we should move on to the next one. Thank you. And I think the next one is Helen. Is that right? Great. Here we go.
Okay, sorry, I lost my Zoom control. Okay, so my proposed experiment is going back to this relationship between the hippocampus and the medial prefrontal cortex, or OFC, depending on which one you want to call it. So I think we're roughly all on the same page that perhaps the hippocampus is particularly involved in a kind of retrieval mechanism during inference, whereas the prefrontal cortex might preferentially represent a category or perhaps an integrated code. The experiment I'm going to propose is really trying to focus on how these two brain regions interact, because I think that if we understand how they interact, that might give us some insight into how an integrated code or a category is actually built in the first place. So, oops. So the study that I showed you earlier was an associative inference task, and in that task, what we showed was that during inferential choice, when we expose the subjects to X, the auditory cue, what we find is that the cells in the hippocampus represent Y in response to X. And we describe this as a prospective code. But what we don't see is representation of the inferred outcome in the hippocampus. And instead we see that in the medial prefrontal cortex and the VTA. And then also what I showed you was that during ripples in the hippocampus, there is co-activation of the inferred relationship, the cells representing X and Z. So the question I sort of have is, what is this activity during sharp wave ripples in periods of rest actually doing, and is it providing a training signal for the representation of an inferred outcome in medial prefrontal cortex and potentially also the midbrain? So I guess I have 4 questions. What is the medial prefrontal cortex actually representing during inference? Is it a conjunctive code? Because it seems to be that it's representing an inferred outcome, but it's doing so in response to this sort of prospective code, potentially. Yeah, it seems like it's doing it in response to the prospective code coming from the hippocampus. How does the medial prefrontal cortex acquire a representation of the inferred outcome? Because in my task and many others, the inferred outcome has not directly been paired with the precondition cue has not been directly paired with an outcome, So there's no sort of known mechanism by which medial prefrontal cortex can acquire a representation of that outcome. The third question is, what is the interaction between the hippocampus and medial prefrontal cortex during rest and sleep? And that might explain the second question. And number 4 is, how do the hippocampal and medial prefrontal cortex representations change with time? Because, yeah, I think we've touched upon this already, but obviously we know that memories in the hippocampus show temporal dependency. So is it the case that there is some kind of transfer of information from the hippocampus to the medial prefrontal cortex with time? Yeah, at the bottom I've just put sort of various references which kind of have inspired these questions. And so one way to try and get at these questions would be to record from particularly the hippocampus and the medial prefrontal cortex simultaneously. And I think to really understand what's being represented, we probably need to do this invasively using electrophysiology, and that will also give us, by doing it in rodents, we'd also then have the control to test causality, but looking at the functional connections between those two regions and manipulating them with optogenetic tools. And we could also have the temporal resolution that we would need in order to look at how ripples in the hippocampus relate to activity in the medial prefrontal cortex. So this is just an example of a recording from those 3 brain regions while a mouse gets a sucrose droplet. But yeah, the idea would be to then record from particularly the medial prefrontal cortex and the hippocampus during an inference task, and to track that recording over time to see how the relationship might change, and also track that relationship during periods of rest and sleep. Yeah, and I didn't show you this earlier, but this sort of data might provide further evidence for the fact that the hippocampal activity during ripples might provide a training signal, because what we see is that those representations of X and Z, the inferred relationship in ripples, occurs in the reverse direction to the inference. So we see cells representing Z, the sucrose, are firing before cells representing X, the auditory cue. And so that might suggest that perhaps by going backwards, you're effectively providing the training signal that you'd need for solving some kind of credit assignment problem. And what's more, activity during ripples is known to be coordinated across neocortex, and by being on a temporally compressed scale, it becomes the temporal scale is concomitant with what you'd require for synaptic plasticity. So you can start to envisage how some kind of representation of the inferred relationship in the hippocampus, which might occur in the reverse direction, might then provide a training signal and induce plasticity in other brain regions, such as the medial prefrontal cortex. Yeah, that's it. Thank you.
Great, thanks. And comments from the panel?
Maybe can I ask you one? So in rodents, when you do this inference task, I remember I think in your initial slides you showed the early and the late kind of phase when they do the test sessions, right? So this must happen over several days, right? So I've always wondered. So I was wondering there if if the mouse is doing the inference between, let's say, the X trying to get to making the inference to Z, but then over time these are experienced basically, because I assume that these are probably the same X, the same kind of stimuli. So do you also have a component that just comes not only from inference but also from direct experience?
So yeah, I mean, so in our experiment, we do it across multiple days because that's the only way to get decent cell yield. But what we do is we try and minimize extinction effects. So X is always presented in the inference test on its own, but because the mice don't get anything in response to X, it's effectively an extinction. So we try and minimize extinction effects by also including like retraining them effectively each day. So we give them extra associative learning between X and Y and conditioning trials between Y and Z before they do the or on the same day as they do the inference test, and that's to try and minimize extinction effects. But yeah, and I think, yeah, I mean, it's messy, right? Like, we don't know exactly what's going on, and we don't know how that retraining is affecting what they're doing. But yeah, I would say also just watching mice behaviorally, I think some of them are extinguishing over time and others are really not. So there's probably a mixture of different strategies going on. But yeah, I think it's an important point to consider.
My question would be kind of about the subfields or the recording site for the hippocampus. So I think that Anna Shapiro bumped into this problem, but essentially when we, there are now number of kind of computational theories about why subfields could be different. And now we also have kind of the idea that things kind of differ along the long axis of the hippocampus and there is functional dissociation there. But whenever we want to kind of like consult rodent literature on that, all what we find is dorsal CA1 because that's kind of where it's easy to endorse the posterior hippocampus analog in humans. So yeah, how difficult now that kind of, you know, we are starting to understand that hippocampus, the dorsal CA1 may just have one set of functional properties, how difficult it would be to start looking kind of elsewhere in the hippocampus, because, for example, if the anterior-posterior story holds, then maybe the prospective code, the XZ representation can be found, it's just in a different part of the hippocampus.
Mm-hmm. Yeah, I think that's definitely an important point to consider. I mean, recording in ventral, which is equivalent to anterior hippocampus, is harder. So that's, yeah, I mean, that's partly why, you know, less people have done it. But recording in CA3 is pretty straightforward, like dorsal CA3 is pretty straightforward. So perhaps that would be the next thing to try, like exploring the other subfields within the dorsal hippocampus in the mouse or rat. But yeah, I think the distinction between anterior and posterior in humans seems quite compelling. So, and yeah, I think that would be something interesting, you know, to look at in the future.
Great. Okay, well, if we can move on, I think that would probably be good. We can save more comments for the discussion right at the very end. But it looks like the next experimental proposal is from Pam. Is that right?
Yes.
Great.
Can you see?
Yep, looks good.
Okay. It's coming.
Great. Yeah, perfect.
So, so we proposed, so for example, the prefrontal cortex maybe can categorize those learned stimulus in one group, then perform the category-based reward inference. So based on this idea, maybe we should have more further question to classify should be considered. For example, so first we should confirm whether PFC maybe is necessary for this kind of category influence or not. So in this maybe first we wanted to make a PFC lesion where impair the performance of the category-based reward prediction, but not performance based on stimulus reward association. So this maybe we should do some monkey study. So next maybe we should ask further. So how do prefrontal cortex generate the category information and execute the category-based influence? So third question maybe we can ask why do the PFC form category representation? So it's depending on the anatomical structure difference or some kind of learning rule or some kind task demands or other else. So I think this maybe I want to try to use some modeling in this sense. So maybe we can easy to control or to modify this kind of structure or use different learning rule. So, but I think maybe the task demands in some sense maybe are very important to whether we prefrontal cortex to form this kind of category representation or not. In some sense, for example, is a very simple task. Maybe you just association or something is enough for to represent the task. But if you have lots of association, maybe category representation maybe has the advantage. Of course, so for our specific question, so we are very interested in how is a newly learned stimulus, for example, new stimulus in our task, ended into the existing category, not to form another new category. So this maybe we can try to use modeling and the monkey study. So another question is how the SR neuron activated in the PFC. So in our idea, so PFC maybe SR neuron maybe have very important to combine the reward information and the category information to perform the inference. But then how could the SR neuron bridge a new stimulus with the reward value? So this maybe we should know the neural circuits in the neural circuits level. So to the information how to combine it together. Of course, from the behavior level, we are very interesting. So how the interaction between the PFC and the other brain area, for example, striatum, hippocampus, to determine the behavior outputs. So in some sense, for example, in some cases, so the output from the prefrontal cortex is inconsistent with the output from other area, for example, striatum and the hippocampus. So which area is more important, or what kind of facts, for example, uncertainty maybe is used to control the behavior. So this maybe, yeah, I prefer to use the modeling. So for the experiment design, so this is from Sakagami's lab. So we designed this kind of task for the monkey. So the monkey have to learn the stimulus association in each group. For example, in this task, in this trial, A1 is presented. After a delay, A2, B2 are presented. Monkey has to choose A2 to get a reward. So we have get 6 types of pairs. For the A1 is associated with B1. If A1 is associated to A2, B1 is associated to B2. A1 is associated A1. A2 is associated with A1. B2 is associated with B2, and so on. So the monkey first, the monkey performs the paired association task. In the paired association task, so the pair changes the block by block. In each block, the same pair is repeated for example, for 40 trials. So then the monkey switch to perform the task we call the match to category task. In this task, pair change the trial by trial. So we should go from first trial A2 is associated to A1, second trial A2, A3 associated with 1, and so on. So in the paired association task, we expect prefrontal neuron maybe just encode the individual association between individual paired associations, so A1, A2. In the match to category task, prefrontal cortex should need to encode the category information or a group of stimuli. So we use this kind of UTAP area electrode or calcium imaging method to record the multi-unit activity in the lateral prefrontal cortex. Here shows some very preliminary example. For example, in this, so actually here shows so at the beginning, the monkey performs the paired association task in this line, so indicate the task switch. After that, the monkey performs the pair to match to category task. So in this paired association task, this example neuron show preferred B1, discriminate B1 and A1 in this association, but do not show any difference between A2, B2, A3, B3. So this may be neuron just encoding this kind of individual stimulus information association. So in this, after the monkey performs the matching to category task, so this shows one example. This neuron prefers B1 in the AB association and the AB2 in the A2B2 pair, and the A3 and the B3 so prefers the B3. So in this sense, so this neuron actually encoding the category information for group B. So they also use calcium images. They show some kind of information. I don't know the detail. So they also can observe this kind of some kind of activity from the calcium image in the lateral prone cortex. So there are further experiments. So after category learning lead to the platinum, we plan to introduce the new pair of stimuli and investigate how they are incorporated into the existing category. We also plan to apply the calcium image to the opposite side hemisphere of the monkey implanted with a Utah allele. So they use two methods to recording this kind of multi-activity to investigate how maybe the category is formed with the older, well-learned one and the newly introduced one. For this kind of modern study, now we try to build a recurrent network with a reinforced learning rule. So we want to try to train the network as the monkey learn this task. So for example, in this model, we have three kind of action. So we have the fixation and the left choice and the right choice. In the model, we have the reward. So then, and also we have the stimulus cue for the A1, A2 choose, and we can choose to which is correct, and we can use this kind of information to train the model. So we want to know after the network learned this kind of task, we want to know what type of neural activity could be found in the network. Of course, whether we can find this kind of SR or R type of neuron in the network. So this is actually want to do if we can find this type of network, so or not. Of course, we can to consider the constraint or the condition to it's the optimal one to form this kind of neural activity. Yeah, this is my proposal. Thank you for your attention.
Great, thank you. Any comments from the panel here?
So if nobody goes second.
Yeah,
so if I understand your paradigm correctly, it's kind of always let's say in your functional categories, let's say the green boxes and red boxes are always on fixed places, and it's always kind of the same response button, right? But you said that, you know, switching hands, for example, you can still get, or others have gotten the abstract code. So I was wondering whether, especially kind of in your model, to really get to the truly abstract code that is neither sensory nor motor, are you planning to, maybe I missed it and you said it, but are you going to try to kind of randomize the side for the response so it the monkey, you know.
So in the for my modeling study or the in which part?
Kind of both, because I feel like you cannot really truly say it's an abstract category code unless you kind of make sure that it's not the response code.
Yes, I think so. In the we actually in those tasks, so we use the association itself. So it's so the position, for example, A1 is presented, A2 and B2. So the position is randomized, I think so. So just the correct association is just the stimulus association. So this is independent on the which position you point. So in this sense, so the important is the stimulus association itself. So in this I think so. For in the experiments, we have two types of tasks. One is paired association. So in this paradigm, so monkey just remember one stimulus associate another stimulus. The A1 is associated with A2. So this kind of in the match to category association. So A1 maybe is associated with A2. A1 is also maybe associated with A3. So when A1 is presented, then monkey don't know whether the coming stimulus is A2 or A3. In that sense, maybe encourage them to categorize to easier for training or for learning or something. So yeah, so this maybe they needed to categorize the stimulus itself.
Yeah.
So yeah, this is also the point.
Great.
Or maybe ask Masamichi if have some other comments or not.
Yeah, I think I agree with you, but I don't know whether it's pure stimulus association, because we asked the monkey to choose one of the stimulus. So maybe through this kind of the experience, so choosing after A1, A2 will be chosen, and after A1, A3 will be chosen in the right or left or something like that. So then maybe some little bit abstract code can be generated. This is not pure stimulus association, I think.
Great, thank you. So if we don't have any more comments right now, I think I'll push us on the time so that we have time to get through the last few little bits of the prepared materials. So we have just a little more to do here from, I think it's Aurelio and Jesse, right? All right, so go ahead.
Okay, can you hear me now?
Yes, we can.
Cool. Okay, so this is an experiment that Aurelio and I had been considering for a while. And I think we've got a lot more to think about how we're going to implement it after we've had all these discussions at this workshop, but I'll tell you what we were planning anyway. So we had an experiment that was designed to test whether generalization is based on memory integration or category inference at learning or at test. And we kind of set it up with the idea that there's some, so if you look at the bottom, you can see memory integration. And there's some studies like those of Shohami that seems to indicate that this happens at learning, while there's other ones that indicate that this might happen at test. And then in terms of category inference, if you consider in the way of Masamichi and Pan, then we still don't know if the categories are being made at learning and then that's just being used at test, or if the categories are actually being made on the fly at test and being used for inference. So we tried to set it up how we can look at these 4 different quadrants. It's a really intensive sounding experiment. The reason for this is we really want to give participants a lot of the novel stimuli that they can make inference based on, because we feel like the second time they see a novel stimulus and have to make an inference, they've already responded to this before, so now they can just use memory. So we really want to see the very first trial when they really have to make an inference. I'm going to walk you through the different days. The first day they'll come in, and I should say this is very much based on the work of Pano Masamichi, but we're going to be doing this with humans. And one of the things too is we just kind of want to test how humans work in this kind of experimental design. So in our case, we're not planning to teach them A, B, C. We just want to teach them some A's lead to some B's. So this is again functional categorization, how we would say it. And we'll teach them a set of A's lead to a set of B's for group 1, and a different set of A's lead to a different set of B's for group 2. And we need to pilot test this. We might need more than 3 or more than 2 in each bit here. We're not sure, because we want to have this for long enough that they're not just going to learn it immediately. We want to have enough trials in the fMRI that we can look at the neural correlates of this learning process. But also, we also want to keep it so that once they've learned it, then they really just do a really boring job where they sit there and do the same thing over and over again, because we want them to overlearn it like the monkeys do. And I think Dasha brought up earlier, maybe it's going to take days for that. And if that's true, then we're going to have to reconsider this a bit more. But we're hoping that we can keep it simple and boring enough that they can overlearn it at this stage. After the people have learned these 2 simple functional categories, we want to test them. And again, this test is very similar to that which Pano Masamichi used. So first of all, they will get some reward instruction trials, where they'll learn that one of the B stimuli from one group is related to a large reward, and a B stimulus from the other group is related to a smaller reward. And then they'll have the test trials. So it's important to note that they don't get to choose which stimulus they see first, because we need an equal amount of trials for analysis. So they're just going to see one stimulus from one group. They don't get to choose that bit. And then subsequently, we're going to ask them to rate, when you see this stimulus, how sure are you that you're going to get large reward? So if you're absolutely sure you're going to get large reward, you should choose 100. And if you don't think you're going to get large reward, you should choose 0. And this is kind of a behavioral measure of inference here. Also, of course, we'll look at the neural activity at the presentation time. And then if they can, if they've made these groups properly, then just like Pan's monkeys, they should be able to choose, okay, this question mark is related to the red stimulus, so then choose the red stimulus and get the appropriate reward for that session. And I also want to mention that we're going to keep these, what we're calling sessions, really, really short. So they might get 2 reward instruction trials and then 3 inference test trials, and that's only 5 trials. So when I'm calling it a session, it's probably a bit strange, but that's the kind of paradigm that it will be. So on the next 3 days, they're just going to come in and learn novel associations. So they've already learned the old stimuli on the left, group 1 and group 2. And now we're going to teach them a bunch of novel stimuli are related to B1, and a bunch of other novel stimuli are related to B2. So if they can use associations, then, I mean, if they can use categorization, they might realize that this means they belong to group 1 or 2, but we're not showing them both of the B stimuli for each group. Then on the 5th day, we're going to do a bunch of different tests. So the first test will be with a subset of the novel stimuli. And basically, so there's an example up on the right-hand, at the top right-hand corner. So they might see a stimulus that belongs to group 1, and they'll see the red group 1B11. So this is one that they've literally seen together, and we're going to ask them, have you seen these together before? So if they can simply recall this, they can say yes. But then in another kind of trial after that, they might see it with the yellow B1 too. And they literally haven't seen these 2 together, but if they've been able to categorize this, then they should be able to say, they might actually have created a false memory, particularly if they've integrated this, I should say. This isn't about categorization, sorry. This is about whether they've integrated this information already or not. And if they have, then they might make the false memory assumption to generalize and say, yes, I've seen this before. And for the other hand, for the 2 B stimuli from the second group, we wouldn't really expect them to say that they've seen these before, because they haven't seen them and they're not related to the same group. So the hypothesis is that if whichever of these strategies is happens during encoding or during the learning phase, then we should start to find these kind of generalization like false memories from this stage. after that, we're going to give them essentially the same task as we did with the other stimuli, where we instruct them that some of the B stimuli, well, one of the B stimuli for one group is related to large reward, and the B stimulus from the other group is related to smaller reward, and test to see if they can follow through and pick the right things to show that they've understood that these belong together. But what's in the kind of manipulation that we're doing here is for these novel stimuli, we haven't shown them with both of the B stimuli for the group. They've only been seen with one of the B stimuli for the group. So then we can start to mess with what we show them with the B stimuli and reward. So take this example here, where at the end, they're going to see this stimulus on the right-hand side that looks like some sake things. And so in the reward instructions in the first session, they might be instructed that the red B is related to big reward and the green B is related to small reward. And in this case, when they see the sake things, they can say, okay, I've directly seen this with the red B, so I can therefore use this to infer that it's related to large reward. Then if there's two ways that we can follow this. So the first one is on the left, further down. And in this case, we switch the reward between the sessions. So we show them the same stimuli, but now we show them that the red stimulus is related to small reward and the green one to large reward. And we think that regardless of the strategy used, they should be able to do this because, yeah, I'll explain why later if necessary. But on the other hand, if we switch the reward, but we also switch which B stimuli that we're showing, then we think we might start to see different results depending on the strategy that's used. So in this case, if you've categorized this new stimulus of the sake into group one, rather than just associate it with the B stimulus that's red, when you're instructed here and you see the yellow thing and then small reward, you can say, okay, yellow belongs to group one, therefore group one is a small reward. This one's also, the sake thing's also in group one, so this is going to lead to a small reward. However, if you use more of an integrative memory kind of strategy here, the sake novel stimulus has never been seen directly with the B12, the yellow stimulus. So this means there's literally no memory in which these two are connected to each other. So then there's no way that they can update this kind of information that they learned in the last trial, where they saw that the red was related to the large reward, so that group one is related to the large reward here, because they have to integrate, if they have to integrate memories, they have to already have a memory between the sake and the yellow reward, but they haven't experienced this yet, so how could they integrate anything there? So we hypothesize that the category-based inference, but not integrative encoding, should allow for correct responses in the second case. And finally, we're going to test them again. It's the exact same memory test with different subset of the novel stimuli. And we just hypothesize that if whichever strategy was happening during the novel stimulus test, rather than during the learning phase, then we should start to find some sort of generalization false memory responses here, but not in the first memory test. So we kind of want to design this experiment to test whether generalization is based on memory integration or category inference. And our hypothesis related to that is that if it is category-based generalization, then the agent should be able to predict reward in both of these two kind of cases that I've explained. And we should find that if we look at the MRI data, the MVPA patterns, when they're in the second sessions here, shouldn't be that different if they're just basing this on category. Whereas on the other hand, if it's a more integrative memory kind of generalization, then we think that the agent should be able to predict reward in the first case, but not the second. And the patterns of neural activity should be completely different in these two sessions. And finally, to test whether this is happening at learning or test, as I explained, we're going to add these little memory tests in. And we think that if the associations happen during learning, then we should find these kind of generalization, false memory responses from the first memory test. Whereas if they were happening on the fly during the test itself, during the inference test, then we shouldn't find them in the first memory test, but we should start to find them from the second one. And similarly, we think that if the associations happen during learning, then the generalizations should relate to neural activity that happened at learning. Whereas if they happened at test, then the generalizations should be related to neural activity that happened at test. And that's all. Thank you.
Great, thank you. So we can have comments on the proposal, and then we can also open it up for questions since we are coming up on the end of the session here. What do we think? Comments? We can also open it up to the audience at this point. We do have one leftover question from before, but if you do have comments on the experimental proposals, please go ahead and put them in the Q&A. And if we have then any closing remarks also, maybe we can move on to that. I know you guys have some closing remarks that you wanted to do, I believe. Is that right? Yeah. So why don't we do that? And then we can close close to on time here. And then what we can do is we can also leave the room open. And so if there are people who are particularly interested in hanging out and talking with the panel afterwards, then you are more than welcome to do that if the panel can stick around. So why don't we go ahead and move on then to the closing remarks if there are no more questions.
So, May?
Yeah.
So I'd like to say one thing about the question about the prototype kind of things. So we looked in an experiment Pan told. So we looked the lateral prefrontal neurons activity for the new stimulus. So newly remembered stimulus. So then we found category information of the lateral prefrontal neuron to the new stimulus is, interestingly, very similar to the category information of the old stimulus. So maybe from the very early, from the very beginning, so category information provided by the lateral prefrontal neuron was very similar to the category information to the old stimulus. That means, so we couldn't find the evidence of the prototype coding in the lateral prefrontal cortex. Yeah, it was surprising. Maybe Pan-san can say something more.
But I don't know why you related the prototype.
Of course, if we have the prototype, so maybe old stimulus is enough trained as a member for the category. But so even a very newly introduced stimulus can provide a very similar amount of category information.
Yeah, I agree. So from this, but my understanding maybe is not so complex, just maybe. So for example, we already, as you maybe suggested, so we already have one association, A1 associated B1 and C1. Then the new stimulus is trained with the B1. So we maybe with 1 day or something. Then we found this the neuron representation for the old stimulus and the new stimuli is very similar. So they have similar stimulus selectivity activity, for example, and also have some similar latency for this discrimination, the preferred one and the non-preferred. So we want to say, so maybe within the 1 day's training for the new stimulus, it's enough to let the prefrontal cortex to encoding the older new stimulus into the existing category. So this may be so I think my idea is like this way. But I still, of course, related to the prototype, I still cannot understand well the relation between the.
I thought it's very simple. If the lateral prefrontal neuron has a prototype, the category information
We never found a prototype in lateral prefrontal, so it was always ventromedial prefrontal. A couple of times, the lateral prefrontal shows exemplar-based category representation.
Okay. Yeah. In that
So
sense.
in that sense, it's actually very similar because that's
consistent with you.
I was thinking that the functional categories may be actually same as the exemplar-based perceptual categories, because what you do is kind of the stimulus category labor associations, and then you really can quickly get from any exemplar to any other exemplars through this simple associative process as well. It's still category representation. It's just exemplar based rather than like the summary prototype representation.
Yeah, another interesting thing is, although the category information was same, but real prediction based on the category was different. So with using the old stimulus, so real prediction information was larger with old stimulus than with new stimulus. So clearly different. Okay.
So I think I'm going to cut this off right now, just so that we can do the final couple slides and formally close out. And then what we've been doing for the other discussions, which have been fantastic, is that if people would like to stay and stick around, then they can. And so we can leave the room open and even pull people onto the stage and have you participate in the discussion. But let's finish up so that we can close sort of on time. And then we can definitely continue the discussion in a more informal format. So I think, Aurelio, you had some slides that were just a few closing remarks. Perfect.
Okay. Can probably see. Can you hear me as well?
Sounds good.
Okay, good. Yeah, so basically just to kind of wrap up and conclude a bit what this whole discussion is kind of has been going around. If we take our initial points of where this kind of controversies in terms and in tasks and in sort of results in a way that we have seen, that might, we might come from different brain regions, and again, learning, generalization response, and a bit again the example of putting humans against animal studies in which we have really well established overlearned kind of association categories, and those that are a bit newer, you might also have functional differences at that kind of level. Maybe a few other things that kind of that I found interesting. Like what the discussion was that we have so of course we have this kind of overlearned behaviors in animals versus humans, but then also of course we have the role of sleep. So and this kind of can help to reinforce those categories, those associations. And one thing that seems really important, rather than just hippocampus or MPFC, is really the connection between those two and how those interact throughout learning those associations, and then later on how they interact at the time of the inference or the generalization. But perhaps another interesting point is how maybe those two areas interact in the sense that if we believe that categories are represented and are used, let's say in the PFC, this should represent some kind of abstract code. And so perhaps it's built through the hippocampus, but then it's kind of, this kind of abstract code are represented or kind of moved to the prefrontal cortex. And other points in which we haven't necessarily touched upon now, we had, but only to some extent, is that, so if we think of those kind of combined memories and more like categorical information, so obviously, and we have talked a bit about that, obviously to create categories, one way could be to combine separate memories. And then once those become, let's say, relatively similar, then you can have a category. But then there's a fine line in between what can be considered category and what is just unstructured kind of sort of integrated memory. And how does the brain arbitrate this? How does the brain sort of knows when to represent a category rather than just keep those single associations? Then as well, and I think this is in general when we talk about the neocortex and maybe the basal ganglia and the hippocampus and so on, the cerebellum never comes up in the discussion. And I guess this is also for various reasons, but I keep on seeing relatively recent papers in which the cerebellum actually has a bit more of a complex functionalities. So that the cerebellum can represent really reward prediction errors. You have sort of category-based activity in cerebellar neurons, and cerebellum itself, in terms of its structure and the microcircuitry, seems to be really sort of well designed to also to extract information and in a way to reduce dimensionality of your sort of your action space. So it might actually be interesting to look at the cerebellum as well for this kind of learning paradigms. And additionally, although a lot of these tasks do involve in one way or the other rewards or sort of reinforcement, these that do not necessarily have to be an actual reward. So for humans, it does not actually have to be money, or for an animal, does not necessarily have to be a drop of juice. It can also be simply motivational or like some kind of intrinsic reward. Like as a participant, you may want to do well in the task, and so just by doing that, by having this kind of motivation, you have an implicit reinforcer. And so I think it's also interesting to sort of start to think a bit about how maybe really the core reinforcement learning machinery interacts with this kind of other machinery that is really devoted to make those inference and those leaps between elements. Because we also know that there is the basal ganglia and the striatum has like all these parallel loops with the neocortex and with the hippocampus as well. So I think there's also something in there that might be worth thinking about. And then I think also, like as Dasha presented, really we might have those parallel generalization strategies. And so at one point the brain should sort of arbitrate between them. So obviously we can sort of define this from the outside based on the, I don't know, on the stimulus property or on some other kind of task properties, but the brain itself has to sort of eventually has to arbitrate, has to define a behavior, has to define a like motor output and action. And whether even if those processes are in parallel, the brain at one point has to sort of to pick which one is the most reliable. And so it might be interesting also to think about that, like what kind of algorithms the brain implements to arbitrate between different generalization strategies. And with that, I think we can just come to the next steps, which would be writing a core manuscript by the group, trying to kind of piece all these bits together into really a like kind of coherent story. And then we'll have commentary pieces, and then ideally, even given the pandemic, we'll still be able to run some experiments, get preliminary results, and which we will be able to present in about a year. And so with this, I would like to thank you all for participating, for the discussion, and for getting here together with this workshop.
Great. Thanks, Aurelio. And thank you all. Great presentations and great discussion. And so now we have the fun part, which is informal discussion. If people want to stick around, I know it's the middle of the night in Japan. But if people want to stick around a little bit, we have a couple people, I think it looks like, who want to be part of the discussion. So we can go ahead and bring those people up. Again, if you'd like to be part of the discussion, go ahead and raise your hand and we can bring you up. And the last thing that I will say as host is this has been really my pleasure, and thank you all for participating in the Generative Adversarial Collaboration Project. We're really excited to have you part of this. And to all of you who are out in the audience, we also still have 2 more of these coming up tomorrow. So if you want to check those out, go ahead and go to the GAC website, gac.ccneuro.org, and you can see the schedules for those there and get the Zoom link as well. So if there are any other folks who would like to be part of the discussion, we can go ahead and bring you up on stage now. Just raise your hand. And so thanks again to our panelists for a fantastic discussion. Jessie, yeah.
I think we need to thank you. You've done a really amazing job. You've kept everything pretty much on time, and you've given everyone a good chance to speak. And thank you very much, Megan.
It has been my pleasure. Absolutely. This has really been fun, and I think there's some great conversation here. So please please continue the discussion or not if you'd like to. And anybody who would like to be part of this also, go ahead and raise your hand and we can bring you up. So thanks everybody. Yeah, and I'll stick around here in the background for a little bit and make sure that everything goes okay. And then if you guys are still going, I will pass off the host baton to someone else and leave you to it. All right, great, thanks.
Thanks a lot.
So we talked a lot. I did talk a lot. So we should let the audience, Brian and Akiel.
I wasn't able to actually attend the full talk. It was I had some meetings and also it was very early in the morning for the California end. So is it more correct to say that you're trying to distinguish the inference process of category versus integrated theory, rather than like the, because I guess one of the things that I commented in my review for the thing was the notion of what generalization actually is defined as in the proposal. So I wasn't sure if you were expanding on that in the workshop or not, but I was just curious of what that ended up being defined as right now.
Yeah, maybe I can start then. So we have, so in terms of what, let's say, what generalization is, as an, as a more like, I don't know, philosophical or so kind of a question. We have to say, so we haven't that much kind of, discussed. We've been going more for the, for discussion on what are the mechanisms that allow for generalization to take place. Generalization kind of intended in a very very straightforward, very simple way, which is to make, to make sort of links or to have a behavioral output based on some evidence that have not necessarily been experienced before, or which only part of the evidence has been experienced before. So it's kind of like a really very simple, I would say, definition in this case, or like operational way of seeing generalization. So I don't know if you have, you have, I don't know, other comments or other ways, because I believe you're coming from a more machine learning perspective.
Yeah, so from a machine perspective, generalization, at least from like a mathematical perspective, is not well defined. We only, at least in the community, have this notion of statistical generalization, which is generalization within distribution, meaning that you could calculate potentially like expected risks and like certain bounds about how poorly you'll do given an evaluation on the same distribution. Now, when you're given something out of distribution, the issue is that that's the generalization that I think most people talk about. And in that case, all bets are kind of off in terms of like what you can say about what is the proper behavior. And at least there is some notion of people working on this in the machine learning community. And this is like in the realm of like causality and how to make things learn like causal representations or causal graphs of like what the actual underlying system is actually doing. But the issue still is that there's this underlying notion of generalization where you can only see generalization after you know what the correct answer is. Meaning that you can only say, okay, well, this thing generalized because I already knew what the right answer was and it did the thing that I expected it to do. Now, this is the weird part about learning generalization, is the fact that you don't know ahead of time what data you need, and the very definition of it says that it's not present in the data. So where would the generalization come from? It'd either be in the already given structure of the model or somehow an association that we didn't expect that exists in the data. So this is kind of one of the big issues, is that like a formal definition of generalization that isn't post hoc, I feel like is necessary, because if you have a post hoc definition, generalization just always boils down to what would a human do? And that's not really a very rigorous or reliable definition, I would say.
Yeah, I felt like we kind of were on the same boat with what we want to study, but we didn't quite agree on what's the big question. I think that from the last Aurelio and Jesse's proposal. I saw kind of why you are making the distinction that you are making, category-based inference versus integrative encoding. I'm not sure whether I agree with it, but I can think about it. Maybe I will. But I think that to some degree, we empirically also do not know what even the question is. So for example, the question for me and Helen seems to be, is it like encoding based, you already build structures, or is it kind of all retrieval based and it's all associative? So for example, there are computational models, let's say for associative inference, like the remerge model, and it does quite well, but then it doesn't explain something. Or in other words, we actually do not know when does this model take place, because it can just as well take place during encoding, right? This inference can be computed at encoding. So when you are looking at like even the definition, the definition you don't like, which is generalization is what the humans do, we actually don't always know what the humans do, because sometimes we make the, you know, the AB or whatever, AC link at encoding. Sometimes we don't. Sometimes we make it a test, but sometimes we don't even do it at test. And it seems, right, like you have to sort of come up with a computational model that can handle all of those scenarios and essentially mess up or not generalize just as often as humans, and do it, sometimes do the links right away and sometimes not do the links until they are needed. And sometimes even when you have all the pieces of information, you still don't make the links even when they are needed. So I think the challenge is kind of like, now we know it's complicated, like can you build a model that clearly has to have more than one mechanism in it and somehow has to resolve, like even if you do this kind of associative based thing, right? Like, is this happening? Sometimes it happens after sleep spontaneously. Sometimes it's demand driven, test driven. Sometimes it's spontaneous at encoding before you even go to sleep. So can you, right? I feel the challenge is there. Like we don't even know really what the humans do, other than we know that it's messy and the humans in seemingly the same situations do at least these three things.
Yeah, I think also one of the other thing that we have often with so if you think of human behavior, but you know, animals as well in general. So when we if you know, if we go beyond like just those tasks that we use in the lab to test these ideas, but if you look more like, you know, at our behaviors in general, like choices that we make, you know, like daily life and so on, it's not always so. It's obviously to some extent we generalize things, but then we also all have this notion of you know, exploration. And so at one point we have that when we consider, for example, we have generalized our behavior to a new situation. This is given by a mix of using some kind of previously whatever sampled data, sampled experiences, and so on, and to some extent as well just, you know, including some kind of stochasticity into it, and just basically go for a new behavior. And so to me that as well is we kind of have a tension in there, in the sense that we, like what Brian says before, you have generalization that, you know, you can define it as like, you know, within your distribution, out of your distribution. But once it's out of your distribution, then so how can you actually explain this? So because you've never sampled any of the data out of the distribution, unless they are, you know, partly overlapping and so on. But if those distributions are just completely, you know, different, then how can you explain that? And I think that's the point, that if you sort of combine, so you combine this notion of previously sampled data, previous sampled experiences, and you combine it with a sort of exploration, now you might be able to sort of, you know, this does not tell me how I would generalize on my first experience, my first trial, my first sample, but following that, then it might.
Right, so I think if you're trying to answer the question of like the category versus like association or integrated based methods of learning, I guess. In that case, the generalization is more of a test of saying, well, the data doesn't give you any sort of clarity on what to do, but the prior and potentially the model you're testing, like a human or an animal, would give you this type of, like, it would come out through that decision that it made. So I agree that that is probably a distinction between those two possible outcomes of like integrated versus category-based, I guess, or association-based versus category. I'm not a neuroscience, so I don't know the terms that well, but like, I think that distinction can be tested in the experiments that you're proposing. I'm not sure if I'd call it generalization as much as just trying to tease apart what's already in the model that would cause it to create a certain behavior that you're expecting to come out.
I never thought about it that way, but now that we are discussing, the other kind of issue is, which I think is an issue, is kind of explaining the integrative encoding, is that there is essentially infinite number, like everything in our real life, right? Everything relates to everything. So what is it that we happen to retrieve during encoding and integrate with the current experience? And to some degree, maybe we need to like start looking into like spontaneous, you know, non-structured recall, because why are we, you know, I can integrate, you know, this session with number of other prior experiences that overlap with this in some way, right? It's a Zoom, or it's a, you know, it's meeting people that I've already met before, or, and so what is it that we end up integrating together kind of proactively? And then at the same time, that could also explain maybe why do we sometimes fail even on demand, right? Like, what is this C related to? And I mean, I have A relates to B and B relates to C. I should be able to kind of compute it on demand, but somehow I failed it. So I'm wondering whether we kind of, in the focusing on the generalization part, we kind of forgot that maybe this is also a question, is what the network spontaneously brings up at any given situation, kind of to a given clue. And that can actually maybe better explain why do we do things. I mean, obviously, plus it's stochastic, but at least matching.
Yeah, so you think, yeah. So basically, you're saying that the way also that we approach all these things when we test them and so on, in a way, is very deterministic, right? So I mean, it's like, you know, you have this item, they are linked together, and that, you know, because you've experienced them, and that's it. There's very little kind of noise or stochasticity to that. And so this is actually something that we've talked about, me and Jess, like the other day, as a kind of also as a potential way that the brain can sort of, you know, even construct a category at one point. So suppose that you're experiencing, you know, that you're experiencing, you know, different episodes, or, you know, different items, different images, and so on. So you have a lot of noise into that. And now the less noise you start to have, or the more kind of defined those links become, then the more likely you are to sort of make a category. On the other hand, you know, if things, you know, if you have a lot of noise in the system, then you might still have a category, but you might have your items, then instead of having, so if the category is well defined, your items are always in the category. On the other hand, you know, if your category is not that well defined, you have a lot of noise in that, then you have items that kind of come in and out. And so stochasticity could come into play kind of at this level. And if you think of it as an integrated memories, I think you have a slightly different mechanism, but in which instead of having those very strong kind of links between different, you know, I can think of this memories, different items, those links are not stable. So, you know, like those associations are not necessarily stable. And so now you start associating new things. And so even when you have, like, you know, on demand and so on, you might not make the right, you know, the right generalization.
Yeah, I mean, your task reminded me of kind of think about stereotypes, right? Like our research little bit kind of speaks to stereotypes, because literally you were rewarded for one member of the category and you are generalizing to the whole category. But you are also kind of hinted that that's what you should do by the experiment context, and you were punished or not rewarded by another member, and you are suddenly like stereotyping and generalizing to the whole thing. There is no kind of guarantee that that's the right thing to do or the wrong thing to do. Is the other, I think with the reward, it's maybe more like that. These are like the good guys and these are kind of the bad guys. But like for the plain associative memory task, yeah, I think that somehow I don't know how easy it's gonna be to do this right computationally, because the context matters. So like, as you say, like because this is all associative network, right? Like which associations will get prioritized, like going back to stereotypes, right? Like if you give Asian women math test, right? You can improve or decrease their performance by evoking one or another stereotype, right? You can either evoke, oh, women are worse in math than men, and their performance goes down, or you can evoke, oh, Asians are, stronger in math than Americans, and then you can improve their performance. And it's this, so kind of like, I feel like that's manipulated by the experimenter, right? But I'm thinking about what do we do in the lab? Are we actually maybe promoting them, or pushing them into evoking one or another prior memory that makes them kind of integrate their current experience with what they already know to kind of conform to that? I mean, obviously we need to start very simple. Hi.
I'm sorry to interrupt. I'm going to go, but you guys stay as long as you like. So enjoy. And
Thank you, Megan.
Okay, bye guys.
I think, okay, so I'm not talking about modeling stuff for a second here, but I just think what Dasha said is really true. Like, I think that the experiment that we proposed is really going to bias people to use a certain kind of strategy. And I think some of the other ones bias people to use different strategies. And so it could be biasing you to specifically link these memories or to specifically look at this category. And it's not that simple at all in the real world. And I think that by the end of this conversation, when we went to propose our experiment, I kind of thought, I still think it's interesting to set up these ways that that we set it up, but I'm not sure. I feel like what we're going to find is almost, we already know what we're going to find because we're setting up the experiment in this way.
Yeah, I'm also like, I'm still wondering why, in the paper has 5 experiments, but I ran like 7 or 8 experiments where I simply, no matter what I did, I wasn't able to replicate that Shohami and Wagner result. And clearly, so, spontaneously, if you just ask people, they don't, just because Peter and Joe likes to both go to the mountains, and you learn that, Peter also likes to go to that grassy meadow behind Brian, it doesn't mean that most people will spontaneously think. Yeah, I mean, it was shocking 80% generalization score. And on trial 1, I have like 52, right? When chance is 50. And then I have that beautiful like learning during test, like exactly what you were saying. I need to cite your stuff if I get to revise it, but it's like, why? I mean, I really stand on my head trying to boost generalization in that paradigm. And, the first time I didn't get results, it's like, what am I doing wrong? But, this experiment 7, this way bigger sample size than was in that paper is like, maybe this is the result, Yeah. So what is it that pushes people to, to kind of do this presumably spontaneous generalization? In associative inference, we ask them to make the inference, but in the acquisitive valence paradigm, in the Shohami and Wagner paradigm, it's supposedly spontaneous.
Sorry. Did you actually manage to get her exact stimuli and use her exact same stimuli?
No, but it's the same. I mean, novel faces, not familiar faces and scenes. And we run it with number of things. We can make people generalize if we manipulate the faces to become more similar.
Yeah, that's what I was thinking with your prototype and exemplar stuff. It depends on the cohesiveness of the stimuli and stuff.
Yeah.
So maybe it's just her stimulus set was like.
I don't think it, because it was randomly paired.
Right.
Yeah. So I think it must have been either her instruction or her, I mean, it was all, you know, Stanford graduate students and her friends who were the participants. Yeah.
And I think that sort of touches on something I was contemplating earlier as well. So I come from like a decision science background, right? So for me, everything's decisions. But then one thing I wonder also today, I mean, they're very nice experiments you guys discussed, but one thing I keep like wondering in the back of my head is where, in a way, it's kind of inverted what we naturally do, right? So normally we make decisions, well, we inform our decisions by generalizations. And I feel having subjects consciously categorize things is maybe in a sense somewhat that unnatural, at least for the non-super smart Stanford students who may actually struggle with these tasks a little bit. And that's also what I kind of wonder with these models. Right? So if you have slightly different task domains, maybe it just also leads to different types of categorization, simply because in real life I don't typically categorize things as much.
Mm-hmm. You reminded me of one study, like I don't know if you know work of Nathaniel Dahl, but he essentially also kind of the decision making, and he thought that he's studying kind of reinforcement learning, right? Like that you accumulate the probabilities and different experiences. And the more he looked into it, the more he found that actually like retrieval of one specific memory, like you make the decision more often based on one specific memory that you retrieve rather than like truly kind of the cumulative probability. So again,
Yeah,
like,
so like,
yeah, we studied in the laboratory and the straight room and the kind of the accumulator and the, you know, the monkey gets this amount of juice and that, like you kind of enforce it in a way, but it, like, and that's what he thought was studying, but then he found out like the model that assumes you actually happen to retrieve one specific memory. And then once I was reading, was like, darn, that's kind of like, although I'm the integrative encoding person, like subjectively, I realized like when I go to a restaurant and I'm deciding, like I can, you know, be pretty crazy about reading the menu as if it was a research article, you know, but at the end, it's kind of like, I remember like, oh, I had this dish somewhere. It's really like almost like episodic memory informed rather than truly.
Yeah, there's also a bunch of like econ and decision science related stuff that's also coming more and more in this direction. But I would say it still brings back a question on why do I think of one thing that I do think of, right? So it's
Okay.
I mean, in a sense, it just raises the question to if I don't categorize or if I don't like go through all my experiences, then the question is like, why that one? Why do I think of the cannelloni I had last week instead of like the lasagna I had last month? Kind of.
Yeah, and I think this kind of sort of also goes back to maybe some of the discussion that I think came in the comments actually to the proposal, and also again that I've heard recently by some colleagues. So if you think of, and maybe we talked a little bit about this with Dasha the last time we met online, in the sense that this kind of propensity, or you know, to use more, you know, those episodes, or more like, you know, to categorize things and so on, is something that changes when you look at psychiatric populations, right? So for example, you know, if you look at I don't know, like at the autism spectrum disorder, for example, or if you look at schizophrenia, they might have actually very different kind of tendencies, you know, along this kind of axis. So in schizophrenia, maybe you try to kind of put things that are, you know, like very quickly you try to create like boundaries and have your like, you know, little categories and so on, and you do this maybe too often. And I think this also speaks to this kind of, so maybe like the normal working of the brain, you might have you might rely, to quite a large extent to those episodes, and you drive with single memories and so on. But then when you start to have it's dysfunctioning, then you start to really create very quickly categories, or binding them together like in boxes and so on. I think it actually might be useful to, in general, to also look at this kind of task, this kind of kind of learning task in those populations, and compare with healthy controls.
I just wanted to ask as a curiosity, is there a canonical, I guess, way that associations are believed to be made in humans? And is that canonical association always very flexible, or is it something that is kind of like a thing that stays around forever? Like there's no context doesn't change it, for example.
So I think that it's kind of the rapid, like for example, single trial association would be thought to be really dependent on the hippocampus. You essentially can't have it associative, like single trial associative learning without hippocampus. And I guess the story is that it functions as an index. So let's say like Brian and the meadow, right? Like I may have a representation of your face in my fusiform face area, the meadow in my hippocampal place area, and they both of them, also these areas have connections to the hippocampus. It's kind of like on the apex of the visual hierarchy. If you ever look at it, right? Like dorsal and ventral parts, they all kind of end up in the hippocampus. And so it has connection to all of this. So it essentially makes the connection inside the hippocampus. So neurons in the hippocampus are the only ones that are flexible enough that can change in single connection in a single trial learning. So can form new synapses. So now when I see a meadow, right, the meadow activates this hippocampal representation, which triggers the, back the memory of Brian, kind of. So it goes through the hippocampus, the association every single time, until if you retrieve it often and often and often, or experience it often and often, the cortex can make direct connections. For example, some of synesthesia is through learning, like the kids had a little, synesthesia is when you see letters and you may see colors, whatever. So one form of synesthesia turned out to be learning related. Like the kids had a little board with the colorful A, B, C, D wooden letters, and they had different colors, and that's what they now see. So because there is now an increased connectivity between like the fusiform board area and color cortex, and the direct connection that simply, anytime you see J, it activates REPT. So there are then cortical connections, so that, the picture of a dog and a word dog now activate, you don't have to go through hippocampus for that association, but before that happens, you have to make that connection.
Like the hippocampal association happens first before it becomes more of like a, almost like a instinctual association.
Yeah, and that's because cortical regions get connected directly. The striatum is another kind of associative area, but it needs a lot more stimulus, and it's usually stimulus response. It's thought to be stimulus response. Like for example, the switching of the hands can mess up the striatum-based learning, because it's literally like, oh, Brian, that's the right button.
Because I think, like, as a AI researcher, this is kind of the associations that I think are missing in the current systems, is the fact that the associations aren't very distant as compared to humans. When they draw associations, the distance is very, very far. Like riding a bike can be associated with driving a car. Whereas for like a deep learning system, it's very greedy. It's like, well, does this visually look the same? If it doesn't, then there's no association. They're completely distinct things. And I think that's why I'm personally very interested in this notion of like how associations are built and what are good priors for associations that can be for very distant things. And it sounds like you're saying that the hippocampal structure is kind of the fundamental component that to create at least the opportunity for those associations to be even learned.
Yeah, so then if we think that the hippocampus is making those associations kind of, so I believe it does it like kind of continuously, then how does it select, which associations are actually there? Because we don't necessarily know, if you can create like, as you were saying before, so that you can create like as many associations as you want in real life, right? I mean, you see constantly things. So how does the brain actually start to select which one are the sort of,
so this is where I think
behaviorally relevant.
Causality is one of the things that like is saying that, okay, well, you can have associations between many things, and a lot of those are mostly spurious, and causality is a game of like eliminating the spurious edges between associations that shouldn't exist. And this is what people are kind of exploring right now, but they're not exploring it from like they already are given the graph and the associations that are possible that exist already. Like I'm interested in the aspect of like, how do you create the canonical association between the nodes? Like how do you create the nodes in the first place? Like the nodes shouldn't already exist.
And the thing is that that's why the hippocampus is so unique, because it can encode these arbitrary associations that are idiosyncratic, and they are going to be useless in the future. But you don't know yet that they will be useless in the future, right? So somebody in the brain takes notice.
Right.
Yeah, that's the point I think really that in the hippocampus and so on, you probably have a lot of these associations that are stored, that are completely useless for maybe for the next 5 years. And then suddenly you have one that is useful. And that I think is actually quite fascinating how those, because in the end it's like kind of like dormant associations, dormant memories, however you want to call them. But then as soon as you have the demand for them, it surfaces. And then you can use it basically.
Yeah, and the other kind of, right, like we also have a neural mechanism for integrative encoding, right? Because whenever you, let's say that while you are looking, you learn AB and now you're looking at BC association, that can remind you, because your B unit in hippocampus is activated, that can remind you of A. So now A, B, and C are all in your mind at the same time, and you kind of update the existing representation. And shockingly, we now have like literally single cell evidence where the people are looking at engrams, and that you reuse the same exact hippocampal engram if things happen soon enough after each other and are similar enough. You reuse the same engram and just update it by this new thing, new piece of information. So that's really, and then you no longer distinguish maybe between these two arenas. In one you got shock, in the other you didn't, but they happened close enough. They are similar to each other. So because you were essentially because of the similarity, you were reminded of the shock that you got in that arena, and now it's all just one integrated memory. So from this perspective, because we have that neural evidence that this is a real thing, like there is no doubt in my mind that also is one mechanism of generalization.
So the similarity you mentioned is really interesting, because I think that is kind of the tricky part, is like what space or what abstraction level do you measure the similarity in? And if you're like, for example, very greedy, like a neural network is, you'll just measure it in like maybe, are these colors the same, or is this visual image similar to this thing? Whereas like similarities for humans and like making associations in a similarity space that's more abstract would say, okay, well, a car and a bike, they're both transportation systems in the abstract sense. And so I can generalize whatever I learned of how to navigate in a car to how I navigate on a bicycle. And I think like those associations that are more abstract tend to, in a human-centric view, generalize better in terms of like we can apply those to more things in the future.
Yeah, that's also something I've been working on. Well, but the problem there is, in some sense, is at least for these computation models, similarity is like very nicely defined. But if you ever try, well, at least also subjectively, we have so many like this looks like this feels very similar. And then trying to parameterize gets incredibly messy, right? Because there's so many different dimensions in reality. So data experience is so incredibly sparse. It becomes like very, very finicky to start working with that. And yet somehow we were able to do it, which is incredibly interesting.
As what people attend to like really quickly, like we have this study, right? Like we have people rate faces, similarity between faces before and after learning, and then they learn something about the faces, and suddenly, you know, if they had the same last name, they start perceiving them as more similar. And presumably other studies too, that neural representation become more similar. And if you have like one well-established and one new piece of information, right? Like say, there was a paper that just came out as an example of Madonna. I don't know how many people still have really strong representation of Madonna, but whatever. So let's say if I found out that Brian is Madonna's boyfriend, right? Suddenly, then my representation, you know, presumably my representation of Madonna is stronger because I for a while. My representation of Brian changes in the cortex to become more similar to my well-established representation because I just met Brian. So yeah, but you can kind of what is similar or what is distinct, you can bias easily by changing the context or reminding people what is important or directing their attention to color versus to the shape, and then whatever they will link or not link based on similarity is difficult to predict. Yeah, as you say. I mean, unless you are really manipulating it in a very controlled manner in the laboratory.
So there is actually in the AI community something that is kind of coming out that is very similar to an associative memory. They call it like a transformer attention model. And this is kind of like the basis of a lot of the natural language success that the community's had. And essentially in that case, it's actually mathematically directly mappable to a hot-field memory, a certain type of hot-field memory. But the idea is that it can draw very distant associations to disparate topics, at least in language. And you can imagine it more like almost like a glorified lookup table that happens to be learnable. And that's probably a very powerful tool just in its own right, because if it's a learnable lookup table, it can learn associations on the fly as it experiences data or is given data. And I think that is something that I think hasn't been explored very much, but I think that explains actually a lot of the success that models are having right now in language, is the fact that associations are very easy to build in very, very large language corpuses.
Yeah. And I think it's also because
Oh, sorry?
I think we are starting to having echo.
Okay. No, I was saying that in those so this is basically those transformer self-attention
right
models. So in which you build and then you can have so I guess you can have, if you have multiple attention nets, then you can start to have as well, you know, like many more of these kind of connections or like, you know, associations between whatever your input is, basically, right, between your channels and so on.
Right. So like the format of them is very simple. It's a query, a key, and a value is the three pieces. And the key kind of contextualizes the values, and the query gets like processed contextually based on what those keys are. And that's how like it goes up the network, I guess.
Mm-hmm.
So what are those called? I didn't catch the name properly.
Sorry?
So that's very interesting. What were these things called?
Those are called transformer networks. They're essentially like there's also another paper that's kind of associates them with hot-field networks. So they're essentially like a certain type of hot-field network that can do very large element lookups, meaning that there could be a very, very large memory space, and you can pull with some reasonable probability like the element you correctly want out of like a very, very large set. But more or less, it's just a glorified lookup table, which I think associative memory is kind of like that too.
Yeah, that's what the hippocampus seems to be doing.
Yeah.
And also like tries to, most of the time tries to keep things separate, or maybe it's doing both things at the same time, right? Keeps them separate in one part, keeps them together, brings them together in another part. I mean, even those studies which directly identify engram, right? Like what's the probability that you happen to stick the needle in exactly the one place where the neurons happen to be? Like essentially zero, right? So it must be, there must be multiple copy, like it's, you know, you don't want one neuron to die in your memory. go
Well,
with.
how distinct are engrams from each other? I mean, I guess they're probably distributed, right, in terms of like the way they're imprinted on like an animal brain, let's say. But the idea would be like, if you saw something similar, would the engram also be similar in some space, or would it be almost completely different set of neurons, differently randomly, like different, completely different areas, even if they're similar? Like, how distinct are things that are at least semantically similar?
But ideally uncorrelated neurons. So I think that's called the pattern separation, which is that's the idea why you can have why you can prevent interference.
Right. Yeah. The interference problem, I guess, is.
Yeah. So it's pattern separation essentially says that the pattern are kind of uncorrelated with each other, irrespective of how similar the inputs are. So it kind of makes similar inputs into distinct representations. That's the story, hippocampal pattern separation. And then there is something else, which is kind of, I think about it as pattern separation on steroids. So this is kind of what you presumably do every time you encode a piece of information. It's sparse, it's unique, and it doesn't depend how similar the inputs are. But there is something that is more recent kind of finding, this pattern separation on steroids that is called pattern differentiation. And it's not automatic as you are encoding things, it's learning based. And it's kind of even more bizarre. The more similar things are, if they have different consequences, or if they, for whatever other reasons you want to keep them separate, they become even more dissimilar. So the memories are literally, the representations are literally less similar than for two things which are completely.
Is it like reward modulated usually, or is it like something that?
I have it with reward, but there are a couple of papers that don't have reward in them. It's kind of like two similar things were paired with the same stimulus. So like a barn with a face and a different barn with the same face, and completely contrary to what anybody would predict. As you go through this task, the hippocampal representations of the two barns become more and more distinct.
And so my follow-up question is, like, as they become more and more distinct, does that hamper the notion of associations between those two things now? Because at least for my work, because I also kind of do things, I have a similar algorithm to this, but the issue is that the more and more distinct they become, the harder and harder it is to actually transfer knowledge from one thing to another thing, because they're not overlapping.
But I think that's kind of the job of this process. So what they did afterwards, which makes it even more exciting, is that they then started pairing these two barns with now two new stimuli.
Oh,
and
okay.
The more distinct people made it, right, to begin with, the less likely they were to suffer from interference between those, right, memories. So it's kind of like so those are like really fascinating study. Mine is reward related for well-run association. So it's kind of a, let's say you have penny, dime, and dollar, and either it was a picture or it was a word on the screen. And people represented, let's say, a picture of a penny versus picture of a dollar as, you know, it's pattern similarity analysis, so fMRI, not single
right.
neuron, but neuron voxels. So the pattern across voxels was most distinct when it was different value, but the same modality, like two pictures. It was less similar than a picture of a, you know, dime to a word dollar. And
I see.
The first time I saw it, I totally freaked out. It was like, this doesn't make any sense. Luckily, the other paper kind of is the Barnes and another paper about the pattern differentiation kind of came out at that time. So I was like, oh, this is actually real. But
it kind of reminds me of, yeah, the Jack Gallant work of like showing that context of the task completely separates out even at the lowest levels of visual representations.
Yeah, I mean, there were big individual differences, but there were also big individual differences in how sensitive people. So it was a memory task. So this was like the cue about how much I'm gonna pay you for remembering this pair.
Oh yeah.
Some people remember the cheap and expensive the same way, and then they don't show the differentiation. And some people have much better memory for the expensive than the cheap Charles, and then they have beautiful differentiation.
Yeah, this seems really tied very much to to this contextual information. So it's a bit like if you take if you compare, let's say, $10 versus $2, and losing $2 versus losing $10, losing $2 in terms of in terms of behavior, in terms of new activity, will look much more like winning $10 than winning $2. Because it's really not absolute. It's completely relative. If you're losing $2 versus losing $10, losing $2 is really good. So this I think this kind of like contextual modulation, I think they play a really strong role, and that's why maybe you also see like if you do this, if you're able to separate this much more, it's also because it's tied to this kind of contextual or task kind of situation that on which the behavior depends then.
Okay, well, lovely discussion. I think
yeah.
I will have to also head out, but
yeah.
I guess Aurelio needs to go to sleep. I don't know where are you guys. So I started at 6 a.m. So now I'm finally awake and ready to talk.
Well, it's time for my dinner, so I'll head off as well.
Mm-hmm.
I'll head off too. I have meetings.
Okay, well, but great meeting you guys.
Yeah, likewise.
Thanks for thanks for sticking around and poking questions into our mind.
Yeah, it was a fun talk and fun presentations. Thank you very much.
Yeah, thank you.
Thanks.
Night.
Thanks to you guys.
Good day.
Have a good day, I guess, to all of you. I'm good to
You too.
sleep.
Bye.
All right. Night.